Hello, everybody, and welcome to the third and final day of the single cell biology and to our session tracking dynamics of single cells. Co-chairing with me this morning will be Dr. Sabrina Spencer from the University of Colorado at Boulder. And I'm very delighted to introduce the first speaker, Rosa Ventotormo from the Wellcome Sanger Institute in UK, who will tell us about mapping the temporal and spatial dynamics of the human endometrium in vivo and in vitro. The stage is yours, Rosa.
Thank you. Thank you for the invitation. It's a pleasure to be here and present the work we do in the lab. So our main question is how the cellular microenvironments are shaping cellular identity and response. And to resolve this question, what we do is to use single cell genomics and imaging tools to profile the tissues in vivo, and then we do computational predictions that we then validate using in vitro models. So central to our main question is the development of computational tools, and particularly this CellPhoneDB, which is a computational tool to look for cell-cell interactions from single-cell data that I developed during my postdoc in the Teichmann team. And what is unique about this method that is based on a repository of ligands and interaction and ligand and receptors and their interactions is that it's a really well curated data set, and also that it comprises these complexes. So, well, we know that the majority of receptors and ligands also are part of have multiple subunits, and you need multiple of them to be co-expressed in order for this receptor to be functional. And this is particularly important one for all the processes, and in our case for development and immune system that we are interested. And that was what is stored in and considered in our data. And basically we then do we then look for ligands and receptors on the single cell data. We were doing random shuffling to look for specific interactions. So moving forward in our team, we are continue developing this CellPhoneDB, and we have recently integrated these predictions with a spatial transcriptomics data, where we just consider that cells that are in the same microenvironment, and that give us the opportunity to improve our interactions. And unpublished data also from our team is a new version of CellPhoneDB, where not only we have improved or store more interactions or more curated interactions, including those that are small molecules, and in this case, because they are non-protein products, we look for the synthetic pathways to build them. But also we have curated several receptors, ligand, and transcription factors that are part of the same pathway. And these give us the opportunity to give us a subset of interactions that are more likely to be happening, because we use this transcription factor that could be predicted by transcriptome, but also by a tag as a sensor of this pathway. So for today's presentation, I'm going to tell you how we have used CellPhoneDB and other tools to study the human endometrium. So, well, we are interested in development, but in this case, we were not looking at the developing embryo, but instead we were looking at the uterus, that is the environment where this embryo develops. So the endometrium is an epithelial tissue that, in comparison to other epithelial tissues in the body, is poorly studied. So this endometrium, this epithelium from the endometrium has this luminal epithelium, will establish communication with the developing embryo, and also have these invaginations or glands that are full of secretions and that will provide the nutrition to the embryo during the first stages of development. So what is quite interesting and got us fascinated about this tissue is the the regenerative capacity. So every month, the endometrium goes to cycles of shedding, regeneration, proliferation, regeneration, and differentiation. And this is mediated by the two ovarian hormones, estrogen and progesterone. So during the first proliferative phase that is dominated by estrogen, then we have the ovulation, and then progesterone levels start increasing, and these tissues start differentiating. And in simian primates, this happens in the absence of embryo implantation. And this really highlights how different this process is, for example, with mouse, and the importance to study the biology of this tissue in humans. So because we don't know that much about this tissue in humans, we ask very basic questions. So what are the cell types that make up the endometrium, and how they change across the menstrual cycle? What are the key signaling pathways that will control this endometrial proliferation and differentiation? And finally, we ask ourselves if we can use the current in vitro models and organoids to validate those. And while we came here from a very basic biological questions, then when we started the work, we also realized how our work will have an impact on women's health, and particular, for example, for endometriosis. I just want to mention endometriosis is a painful disease that is suffering 1 out of 10 women in reproductive age. And it's quite surprising that there are not good or non-invasive diagnostic tools and either treatment for these women. And in part is because we have a very poor knowledge about the biology of the tissue. So the first thing we did is to profile samples. And in particular for this cohort, what we were sampling is not only endometrial biopsies that takes the superficial layer, but also full uterine samples that came from the organ donor program. So people who die from non-gynecological disorders. And then we profile our 10 individuals with other individuals that were published recently in a Nature Medicine paper. And then we integrated all the data sets and defined the main populations. So we managed to define a lot of main populations and subsets. And just as a side story for this, I'm quite excited because in the immune part, we defined the 3 NK cells that I previously defined during my postdoc in the uterus during pregnancy and interactions with the placenta. But this is not that was anecdotic, and I don't want to distract you on that, because for this work, we are focusing on the epithelial cells. So this tissue, it changes a lot during the proliferative and secretory phase. And as you can see, the composition is changing. So in the proliferative, we have these SOX9 populations, which we define 2 populations depending on the expression of LGR5. And then we have in the more secretory phase or differentiated phase, the luminal, the glandular secretory, and the ciliated. So because we define these SOX9 populations that depend on the expression of LGR5, we then wanted to see where they are located. And to do that, we use spatial transcriptomics, and then we use cell to location to integrate it with single cell and deconvolve the wells. And what we can nicely see is that the LGR5 populations are on the lumen, while the LGR5 negative populations are in the basal layer, and we suspect that these luminal ones are giving rise to the populations in the lumen, while these ones will be more to the ones in the glands. And we have also validated these using high-resolution microscopy. So with this map, this is all in healthy samples, and then we wanted to see if we can use that information to understand better endometrial pathologies. And basically we analyze endometrial cancer and also endometriosis, and in both cases we see an enrichment of the SOX9 positive population. And just this is also quite a nice finding in the endometrial cancer, because we have two types, the serous and the endometrioid. The serous is more aggressive, and we see the signature. This is just the convoluting bulk data with single cell, and in this case, we see the signature of LGR5 positive progenitor, while in the endometrioid endometrial cancer, we see that half of them are LGR5 positive, while the other half are LGR5 negative. And actually, the ones that are LGR5 positive is correlating with more advances of this cancer. So with that, I'm just highlighting how these pathologies are enriched on the progenitors. It really highlights how important it is for them to have these epithelial cells to properly differentiate. And that's where we started to look for the differentiation pathways by measuring the transcription factors and also their activities just by looking at the genes that are known to be regulated by these transcription factors. And then what we see is that in the epithelial cells that will differentiate towards the ciliated lineage, the Wnt pathways become active, while the ones that differentiate to the secretory lineage, it's an inhibition of Wnt and then an activation of Notch. We wanted to see if this pattern of the pathways is also seen in space, and we do that again by using special transcriptomics. Here we didn't integrate it with single cell, we just did clustering, unbiased clustering on the epithelial cells, and you can nicely see how just the unbiased clustering really clusters with the lumen and the glands and the base, are just highlighting how important is the environment for the identity of those cells. And again, we see the same pattern that more in the lumen, where it's enriched in ciliated cells. We see Wnt inhibitory signals, while in the glands, that is more enriched in secretory cells, is more transcription factors that are known to be inhibited by Wnt and transcription factors that are known to be active by Notch. And we just also validated signals for Wnt and Notch just using high resolution microscopy and quantifying, and just here showing the high presence of Notch in the glands in comparison to the lumen, and also it is quantified here. So we of course wanted to study the signals from Wnt and Notch, and to do that we use CellPhoneDB, the one that is integrated with the space. And we divide it in proliferative and secretory phase. And well, in the proliferative phase, we see that Wnt is active kind of everywhere, and that makes sense because it's essential for proliferation. But then in the secretory, in the cells that are the secretory lineage and the ones in the glands, they downregulate Wnt receptors, and also these stromal cells or endometrial stromal cells that are fibroblasts that intermingle with the glands express DKK1, which is a potent Wnt inhibitor. So with that, really from this single cell profiling, what we see is this disbalance between Wnt and Notch happening in different spaces of the tissue, in the glands, on the lumen, where it's enriched on different epithelial space, the secretory or the ciliated. So we wanted to really validate our findings and understand how this disbalance between Wnt and Notch can affect epithelial cell differentiation. And to do that, we use endometrial organoids, together with Margarita Turco from Cambridge University. And what is quite nice from these organoids is that you derive them from primary tissue, and then once you have established them, you can stimulate them with hormones, with estrogen and progesterone, and they behave in theory as the in vivo epithelial cells in the endometrium. So we profiled them because the first thing was to benchmark the model. We profiled by single cell. By markers, we can annotate the different ciliated and secretory lineage. But of course, we wanted to kind of benchmark it in a more quantitative way. And to do that, what we did is to build a logistic regression model using our in vivo epithelial cells, and we project it into the organoids. And what we see is that, well, just want to mention here, we just consider the cells that are induced by hormones, because in vivo you always have hormones. And we can nicely see that upon estrogen, we have the SOX9 populations. And then when we put the progesterone, we have the ciliated lineage popping up and these preciliated populations that we identify. And then we have the secretory, and in the secretory lineage, we have some SOX9 that are becoming differentiated into the secretory one. So we don't care, to be honest, that much if the organoid is not fully differentiated or not, which as you can see in the secretory is not fully, it will be around 40% of the secretory cells. But what we care about is if those epithelial cells are responding in the same way that they in vivo, because we want to understand the pathways for differentiation. And to do that, again, on these subsets, we calculated the transcription factors and their activities, and we could nicely validate how the same transcription factors that we show in vivo that are responding in the ciliated lineage pop up here, and exactly the same happens with the transcription factors for the secretory lineage. So therefore, we have a nice system where we can validate the effect of Notch and Wnt, and that's what we did. So here, in addition to put the hormones, we combined them with 2 inhibitors for Notch and 2 inhibitors for Wnt, and we performed single-cell transcriptomics data on different conditions, and in addition, we do some immunostaining. And I'm just going to show you how nice the phenotypes were. So basically, when we put the Notch inhibitors here, we can nicely see how we don't have secretions at all, and then we have a lot of ciliated cells, and this was also recapitulated by a single cell. And actually, the completely opposite pattern happens with the Wnt inhibitors when we have very few ciliated cells and we have loads of secretions. So then the nice thing about profiling with single-cell transcriptomics is that in addition to looking at the efficiency, which was really validated for the Notch and Wnt, we can also look for the accuracy. And that was relevant in the condition where we put Wnt inhibitor, because for the Notch inhibitor, we don't see a new subset coming up, but we do see a new subset in the Wnt inhibitor. And actually, we wanted to see, is this subset producing more secretion? And that's what we did here. And actually, you can nicely see how this is the case. So when you have hormones and Wnt inhibitor, then you have more secretions. And we validated this also at the level of protein. And this is in vivo, is happening because you have the stromal cells that will induce this Wnt inhibitor environment on top of the progesterone. And then we can also dig more into the mechanisms and the relationship between hormones and pathways. And for example, one of the things that we realized that Wnt inhibitor, if you put it on their own, it doesn't induce the secretions. And this is just because the hormones are required to, for example, activate Notch that is not activated if you only put a Wnt inhibitor. And with all of that, we have built this model using single cell and spatial methods and this nice resource that is publicly available, and we have a nice interface to visualize it, where we can see how cell stage change across the menstrual cycle, and then we also define these 2 environments that have a disbalance between Wnt and Notch. And well, that's all for us. I just want to acknowledge the 2 members of my team, Luz Garcia Alonso and Louis Francois Handfield, who led this project. This project was also a collaboration with the Sarah Teichmann team. That's because I started this project while I was a postdoc in her team. And also with Omer Bayraktar for all the help in the imaging, and Margarita Turco for the help on the organoids, and of course the donors. And thank you so much.
Thank you, Roder, for a super interesting talk. We have a question from Arjun Raj. Very nice work. It's great to see the environment and effects of space. Do you think the reason for the differences has its origin in differences in the protein composition of the ECM, or by physical differences, like different tensions on the cells in these different locations, or something else entirely?
You mean, sorry, between the organoids and the in vivo data, or?
I'm not sure between the organoids or between the different stages or between the different zones, like the ciliated zone or the secretory zone. So he's wondering about the role of the ECM tension versus maybe morphogens or.
Yeah, that's correct.
I hope I get it right, Arjun.
Yeah, we of course simplify it to the Wnt and nodes, but there is quite differences because, for example, in the lumen, you don't have that much of a connection with these stromal cells where you have the glands embedded with, and that really it's not only the pathways, but of course also the extracellular matrix proteins. But we have just reduced it to this Wnt and node signaling. But yeah, there are also a lot of different interactions that we haven't explored so far.
Thanks.
Okay, so the next question is from Yotam Harnik, who says, thanks for the interesting talk. How do you think the balance between the two lineages is regulated?
Well, yeah, I do think that is really because the spatial position that you have into the space, because, well, and that's why it is a balance. It's not that in the lumen you only have ciliated cells and in the glands you only have secretory, but because in the glands you have more access to a Wnt inhibitor environment, then you can really differentiate towards the secretory lineage, while in the lumen, where you don't have this access to that much of a Wnt inhibitor, then you differentiate towards the other lineage. So, but again, it is a balance because it is really an efficiency thing. It's not that it's black and white, if that makes sense. So it's really the spatial position that I do think that will change, and the interactions that this spatial position is associated with.
Thanks. We have another question from Maria Bermudez. Do you know if this Notch-Wnt signaling relationship can be changed in pathological conditions such as cancer?
Yeah, I mean, that's a really interesting question and something that we have been also exploring. And for example, we have been looking for GWAS, and we do see on the GWAS a lot of hits really are related to the Wnt and Notch, and also the crosstalk between the fibroblasts and the epithelial cells. So certainly, I'm pretty convinced that there will be something related to that. But of course, we will need to explore further using pathological samples. So this is just crossing the GWAS hits with our powerful kind of defined single cell data set from healthy samples.
Okay, one more from Joshua Thiessen. Have you performed RNA velocity analysis on your data? One of the first UMAPs you showed had a somewhat circular appearance, similar to the cell cycle UMAP shown in one of the talks yesterday.
Yeah, I mean, the problem with velocity for the organoids, yes, and we can reconstruct it easily. The problem with the adult samples is that they come from a lot of individuals. So it is really difficult to build trajectories on them, if that makes sense. And using velocity and all these tools. So yes, for the organoids, that's simple. But for the in vivo data, it is, and we have also done clonal organoids, so derived from one single cell. So we are sure that this progenitor can differentiate into both of them. But for the in vivo data, yeah, it is, because you never have the progenitors and the differentiated cells on the same individual. It really changes, and that's why it is really difficult. I wouldn't say impossible, but at least in our hands, it was almost impossible.
Okay, there's a very interesting question here from Timothy Powell. Is there commonality in gene expression differences between the ciliated and secretory epithelial cells in the endometrium and these epithelial cells in lung or intestine? Can you see a common signature for ciliated cells in different locations or tissues?
Yeah, that's correct. We haven't done a comprehensive analysis, but actually they are quite similar with the ones in the lung. We have done some analysis, and you do see a lot of similarities between the epithelial cells in the endometrium and the lung, and also this Wnt and Notch regulation is similar. It's not exactly the same because some stem cell populations you see in the lung, you actually don't see them in the endometrium. So not exactly the same, but a lot of common pathways on the ones in the lung.
Okay, I think we have about 1 more minute, so let's squeeze in one more question from Jingqi Wang. In the cell phone DB, could you quickly explain how you take into the concentrations combinations of ligands and receptors to predict the active pathway? So how do you take into account the concentrations combinations of ligands and receptors to predict?
Okay, I'm not sure I am following that question. So do you mean, so we just consider, in CellPhoneDB, we just consider the expression of receptors and ligands. And what we did is, well, the point is a lot of ligands and secreted ligands we have done, they are very expressed by very few cells. So that's where, I mean, the user can decide, but we usually say that around 10% of cells need to express that. And then the random shuffling, what allows us is to look for a specific, but actually you can also run the same, and we are running the same with differential expression genes to prioritize, but this is more to prioritize the interactions that are specific. I don't know if that's what the question was.
I think that makes sense. Okay, well, thank you, Rosa. I think it's time to move on to our next speaker, who is Sylvia Santos from the Francis Crick Institute in the UK, and she'll be talking about signaling dynamics for cellular fate control. So go ahead, Sylvia.
Thank you so much, Sabrina. Thank you so much to Arjun and Shalev for the kind invitation to speak. This is has been such an exciting couple of days, and I would love to be in Banff with all of you and actually have one-to-one interactions. So my lab is very interested in understanding how cells make decisions. I'm trying to change my slide. Just 1 second. Perfect. So our lab combines experimental and computational approaches to understand cell decision making. And in particular, we've been very interested in understanding 2 fundamental decisions: the decision to undergo cell division and the decision to undergo cellular differentiation. We are very interested to understand how these 2 decisions are ultimately very interdependent. Now, an important concept in cell decision-making is the idea that is very rare in biology, that a single gene or a single protein alone are responsible for cellular responses. So on the contrary, oftentimes it's really the ability for molecules to interact with one another, and ultimately on how the concentrations and how the activities of these molecules are regulated in time and are regulated in space that are really key for cellular responses. I think cells do this best by coordinating regulatory networks in ways that allows them to discriminate between different signals and allows these networks to trigger different cellular states and ultimately different fates. So an extremely useful system to explore regulatory principles in decision-making are actually human embryonic stem cells, which have this magical propensity to differentiate into the 3 germ layers and virtually give rise to all the cells in the human adult body. These cells, of course, also self-renew, so they are also an awesome system to understand cell division. They are incredibly trackable and a fantastic system to understand how cells compute decisions and give rise to early fate choices during human development. Now, the model we use in the lab is mesendoderm differentiation of human embryonic stem cells, and this is driven by one molecule called BMP4. And upon BMP4 stimulation, these cells go from these tiny cells that grow in colonies to cells that are much, much bigger, about 10 times bigger, and are importantly able to migrate away from the colonies and live as single cells. Now, BMP is really a fantastic system to use here because not only we differentiate cells into mesoderm, so BMP is known to be a mesoderm factor, but actually about 3 days after BMP stimulation, we recapitulate really gastrulation. So we see populations of mesendoderm cells, of mesoderm and endoderm, and populations of extraembryonic layers. And so in a way, this is a very important system to mimic gastrulation in vitro. But what makes it extremely useful is that we can actually use micropatterns and thereby confine these cells to fixed geometries. And when we do that, and this is, by the way, a technique developed at the Rockefeller Institute by Aria Wormflash, magic really happens. So what you see is the exact patterning that you see in a human embryo, where the outer layers are the extraembryonic layers in the embryo. The middle layers are really the mesendoderm. So we've got endoderm and mesoderm cells, and the inner center of these colonies are going to give rise to the ectoderm. So exactly as you would see in the human embryo. So we can now combine, of course, our mycopatterns and doing imaging in them with single-cell RNA-seq data, and have this magic information of where each gene and each protein actually physically are. So we're having, a fantastic spatial and temporal resolution of these first decisions in human development. So, of course, to combine sequencing and imaging, we also need a lot of image analysis. And in the lab, we have been in recent years putting a lot of effort to use neural networks and track these cells. Mind you, these cells change dramatically in morphology. And so we are now able to track individual cells within these colonies as they have these dramatic changes in morphology. So let me tell you a little bit about what happens during BMP differentiation. So the changes in morphology were obvious. I showed you in the first movie. There are, of course, massive changes in gene expression programs where pluripotent genes are downregulated and lineage-specific genes are upregulated. There's global changes in epigenetics and the cell cycle remodels, whereby differentiated cells have much slower division cycles. Now, all of these are actually late events in differentiation, most happening about a day or two since each of these pluripotent cells sees BMP for the first time. So you basically need days for any visual changes in gene expression or in chromatin or in cell cycle regulation. So Alex, a PhD student in my lab, was very interested in understanding whether irreversible commitment to differentiation was also a late event, was also something that happened as cells progress through differentiation. And what Alex found was actually extremely surprising. And it was the fact that cells actually commit to differentiate within minutes. So the experiment that Alex did was very simple. She pulsed cells with BMP for just half an hour. And what you can appreciate, hopefully, here is that 3 days later, these cells have changed in morphology. They have upregulated these red genes here, so the mesendoderm-specific genes, and downregulated pluripotent genes, exactly the same way as cells that have seen BMP in a sustained manner for 3 days. Now, if the pulse of BMP is any shorter than this half an hour, absolutely nothing happens. The cells continue proliferating away, and all the pluripotent genes are upregulated. So she saw this, of course, for a variety of other genes, but importantly, when we use our mycopattern system, you see that the pulse of BMP gives rise to exactly the same signaling, exactly the same first waves of gene expression, and exactly the same hallmarks of gastrulation that we see when we stimulate with sustained BMP. So this was extremely exciting. Now, how are these signals decoded by the cells? So the BMP is actually transduced by the so-called SMAD transduction network. SMADs are transcription factors. They have to go into the nucleus to transcribe genes. And if you don't have SMAD, even in the presence of BMP, these colonies just continue proliferating away and the cells don't differentiate. Now, we noticed that SMAD activation was sustained after BMP stimulation. But very interestingly, it had this switch-like behavior where the cells at the end, at the periphery of the colonies, got activated first, and then with time, more and more cells got activated. This switch-like behavior is also seen as we look at SMAD activity as a function of BMP concentration, and you can see that if you add a little bit of BMP, not much happens, but after some threshold concentration, you clearly see that more and more cells jump into this SMAD activated state. Now, switch-like responses and bistability are common in cellular transitions, and for the most part, they give rise to actually irreversible responses. And indeed, we see this. We see that if we activate SMAD pathway with BMP at maximum activity, even if you remove BMP to 0, these pathways remain on, showing a degree of irreversibility. Now, why irreversibility? Well, part of the answer is that these pathways are actually embedded in positive feedback regulation. So we found that one of the first genes that SMAD regulates is actually its own activator, the BMP receptor itself. So classical example where activator activates activator. But how are these signals actually decoded? So how is this upstream dynamics of signaling pathway actually translated into ultimately commitment to differentiation? So we ran a RNA-seq experiment, and one of the genes with the highest score was actually not known to be a SMAD target gene, and this is this gene called GATA3. So we looked into it a little bit. So it was very interesting to see that GATA3 expression was very early, right? So at the protein level, after 2 hours of BMP, we start seeing cells expressing GATA3. But importantly, it had the same periphery versus inside of the colony, so the same spatial activation or expression as SMAD activation. And in fact, every time we see GATA3 at the mRNA level, we saw expression or activity of SMAD. The same dose of BMP that is important to activate SMAD is exactly the same that leads to expression of this gene. So these early genes are actually in not only GATA3, but in this little cluster of genes are actually mirroring completely what happens in upstream signaling. So that was very interesting. So we see that GATA3 is also embedded in positive feedback. So this is a chip experiment where we see beautiful peaks after BMP stimulation on GATA3 itself. And in fact, these nested feedbacks that we uncovered are predicted to help maintain a memory of this signaling and this expression of early genes. So as you activate the pathways with a little bit of BMP, even if you remove BMP to zero, these signaling pathways are going to be on, and the same with expression of GATA3. Now, positive feedback is obviously a common motif in cellular transitions, and so we were excited to see that potentially this is what gave rise to a switch-like irreversible transition and commitment to differentiation. So we were very excited about these dynamics, but we really wanted to understand whether GATA3 could actually be an early commitment gene that would tell us at the single cell level that cells had committed to undergoing differentiation, exiting pluripotency irreversibly days before we actually see any loss of pluripotency or any upregulation of lineage-specific markers. So this was very exciting for us to explore. What would you expect from an early commitment genes? Well, potentially, when induced, they should induce differentiation. This is exactly what we see. So we induce GATA3 in the absence of any differentiation cues, and we see upregulation of differentiation genes. And interestingly, we see this multi-lineage expression, very, very interesting for us. Now, conversely, if we knock out GATA3 with CRISPR, we see a delay in differentiation. So here's just a handful of genes cherry-picked to show mesendoderm differentiation, and you see that basically there is very much delay or a non-expression of genes in the knockout situation. So in addition, we expect an early commitment gene to regulate early targets. Now, this is a ChIP-seq over time of GATA3, and we see the common expected late targets of a fate marker like GATA3 used to be known for. So this means that, in early time points, you don't see GATA3 binding to any gene, and then you see these beautiful peaks after 48 hours. But actually what we see is that in about 3,000 genes, we actually see a very early regulation. So 6 hours GATA3 is sitting on these genes, and after which this regulation is lost. So GATA3 is definitely regulating genes very early during differentiation. And there is, the potential for network rewiring, which is extremely exciting. So we image, of course, a knock-in of GATA3, right? And what I wanted to show you, this is just I'm showing you the movie in a sec, but this is just a still image of a 9 hour. And you see that in this colony, every cell, every precursor cell is expressing GATA3, potentially at different levels, but I'll come to that. So initially there is no GATA3, we stimulate cells with BMP, and you start seeing, the cells spreading out, changing shape, and GATA3 late, just really expressed especially in these flat cells outside of the main colony. So we actually see that the levels of GATA3 are predictive of fate. So these extra embryonic layers, so the cells, the flat cells in the outside of the colony, are going to correlate very nicely with high GATA3 levels. The inside of the colony anti-correlate very nicely with GATA3 levels, so no GATA3. But very interestingly, when you have intermediate levels of GATA3, you actually specify mesendoderm. So it can be basically predictive of fate choice. So this is an example of just single cell tracing. These cells here have no GATA3. We specify, we have an intermediate level of GATA3, and we have a high level of GATA3. And what you can see is that the marker for mesendoderm, which is Brachyury or TBXT, is really only expressed in these intermediate GATA levels. So one important thing we found out along the way was actually GATA3 is a pioneer factor that opens chromatin early in differentiation. So this is a complex slide, so I'm just going to try to lead you to this point here. So this is basically ATAC-seq. What ATAC tells you is that the chromatin is opening, so at 0 hours and then 48 hours, 72 hours. And we combine this with CHIP and see that GATA3 is actually sitting at this chromatin later. Now, if you knock out GATA3, you see that this chromatin doesn't open at all. Okay. The reverse is also true. GATA3 is able to close chromatin. So this is ATAC-seq again. So differentiation in this axis, you see that the chromatin is closing. GATA3 binds initially, but then doesn't bind anymore. And if GATA3 is not there, the chromatin doesn't close. It's very open. So we are super excited about this. This is basically just to show you in all the peaks. So these early peaks of GATA are basically sites where the chromatin tends to decrease, so tends to close. And this is perturbed when we don't have GATA3. Okay, so I'm basically trying to tell you that potentially we have found a new class of genes that are able to tell us way in advance that the cells have committed irreversibly to differentiate. And one of these genes is GATA3. So we think that it's behaving as an early commitment factor. If that's true, then this first experiment that my PhD student did, Alex, with a pulse of BMP, should basically be potentially compromised if we don't have GATA3. So this was the experiment. A pulse of BMP gives rise to differentiation. But when we don't have GATA3, this is absolutely compromised. So showing that GATA3 is very likely to be an early commitment gene. So I would like to finish by asking one last question, and it is, I'm basically showing you that cells irreversibly leave pluripotency and commit to differentiation. But how irreversible is actually this commitment? In other words, could we divert fate choice or not in this scenario? So the experiment that we did was very simple, and it was giving a pulse of BMP to the cells. And instead of putting the cells back in pluripotency media, we actually triggered another pathway. So we challenged the cells with retinoic acid for them to differentiate into ectoderm. And let's see what happens. So can the cells divert fate, or once they've committed to a fate? So cells that see BMP in a sustained manner become mesendoderm. Cells that see retinoic acid become trophoectoderm. A pulse of BMP, cells don't care, they will become mesendoderm. What happens now when we try to switch fates? And the cells are able to switch indeed. And this actually happens within a window of time of about 48 hours. So what I tried to show you today was that commitment to differentiation in human ES cells is an unexpectedly early event. We see that positive feedback in the networks that transduce these signals is really important to maintain a memory of these same signals. And we see that early genes, specifically a cluster of genes, really mirror the dynamics of that upstream signaling network. And we think that GATA3 might be an early commitment gene. Lastly, I showed you that while cells irreversibly leave pluripotency, they remain plastic to fate choice. And we are at the moment very interested in understanding what controls this plasticity with this plastic period. And just to give you an outlook to close, I think there has been a couple of models proposed for development, for fate choices during development. And the classical one has been very much influenced by morphogen gradients, where the position of a cell within the embryo really determines how much concentration of the precise signal they see, and therefore the pathway down in differentiation is very linear. Now, there is another model, and this has been very, very well shown for hematopoiesis, which is the model of cellular plasticity, whereby the first precursor cells are actually plastic cells. And with time, a fate is chosen and other two fates are repressed. And this is where we find basically the late commitment genes, or the fate genes that everybody knows. So we think that our data is potentially quite in line with this second model, whereby early on we have early genes that actually make the cells plastic to fate choice, and that with time then one fate is going to be selected. And with that, I would like to finish by thanking, so all of the first work that I've shown you was done by Alex, a PhD student in the lab. She's no longer with us, unfortunately. And the GATA3 early commitment as an early commitment gene is unpublished work done by Adrian, a fantastic postdoc in the lab. I would like to thank collaborators and the CRIC for being a fantastic environment. And thank you very much for your attention.
Thank you very much, Sylvia, for the wonderful talk. We have a question from Kaiser Loel. Great talk. Do you think that GATA is directly repressing the sites that are closing during differentiation, or do you think that it's some sort of indirect effect?
Thank you for the question. Potentially both. So we see very interestingly that one of the genes, or one of the set of genes that GATA regulates, are ectoderm genes, for example, PAX6. So it's basically promoting the mesendoderm branch and really repressing ectoderm genes, so not letting the cells become a fate any longer. And that at that site, we see beautifully that the chromatin is closing with time for ectoderm genes. So in that sense, is very direct. In other examples, we don't see as much as a direct cause. We are at the moment trying to, we have identified a set of candidate genes that we think might be binding partners, important for closing and opening chromatin. And we are basically knocking them out and seeing what happens to chromatin state at these sites.
Okay, thanks. Next question is from Dagan Segal, who asks, do you think you could model symmetry breaking with this system as well, for example, through asymmetric micropatterns?
We have explored different shapes of the micropatterns a little bit. I do think that probably the best way to go forward is using the new 3D system that was developed by Naomi Morris and Alfonso Martinez Arias, which is, of course, the gastroloid system. So human gastroloids are now available. I mean, we do them in the lab quite routinely now. We find that there are weaknesses in terms of really representing human development, but I think if you are interested in symmetry breaking, this would be the best system. And you've heard Prisca Liberali talk, and I think their lab is fantastic, has been doing fantastic work on this. So I would go for the 3D system in here.
Okay, thanks. There's a technical question here from Di Hu. How many cells did you use for your chip, and how many sequencing reads did you use? The browser shot you showed looks very clean.
Yeah, thank you. So I don't know by heart, but we published cell protocols, STAR protocols. So please check us, because we noticed that a lot of people, even like very well-established labs of human development, were basically coming to us to ask for advice on how to do these chips. So we decided just to publish and make it available for everyone. So check it and please email me if you have any questions afterwards.
Okay, another question here from Jiali. Excellent talk. How are the other early progenitor markers such as the IL-1, CREB-PA changing by adding DMP4 or in the knockout of GATA3?
Yeah, so ISL1 is a direct target of GATA3. GATA3 is binding there at 6 hours. So you would expect this gene to come up after BMP stimulation, but in fact, you see a very big delay when GATA3 is not there. So it's actually upstream. So this gene is downstream of GATA3. So thanks for the question.
Okay, I'll just ask one last question of my own. Did you try modifying the size of these micro patterns? And are there any interesting changes in the spatial pattern when you impose different sizes?
Yeah, so I mean, there is really some type of a sizer, right? So if you have tiny micropatterns, you will form just the outer layer. So what we think are the extra embryonic layers. I mean, people now think that is basically amnion that we are forming. If you increase a little bit, then you start forming the outer and the immediately intermediate layer, and only a specific size will form the 3 layers. So the size makes a huge difference. The mechanism of this sizing, we actually don't know.
Okay, thanks very much, Sylvia. We'll move on now. Our next speaker is Sabrina Spencer from the Department of Biochemistry, University of Colorado, Boulder. And she'll tell us about real-time visualization of rapid escape from BRAF inhibition in single melanoma cells. Thank you, Sabrina.
Okay, great. So first off, thanks to Shalev and Arjun for inviting me to this great meeting. My lab specializes in long-term time-lapse microscopy to study cell proliferation at the single cell level. And similar to Arjun, I will also talk about melanoma drug responses, but in this talk, we're going to focus on just the very rapid initial quick responses that cells, melanoma cells, undergo in response to BRAF inhibition, and how cells can actually escape from drug in just a matter of a few days in a non-genetic fashion. So, of course, proliferation is driven by the MAP kinase pathway, RAS, RAF, MEK, and ERK, which drives cell cycle entry. And over 50% of melanomas are driven by an activating mutation in the BRAF kinase, which drives hyperactive cell proliferation. About a decade ago, some great BRAF inhibitors came on the market, which very effectively block proliferation from this mutant oncogene. And they have very dramatic and positive effects in patients, at least initially. But what almost inevitably happens is that some cancer cells find their way around the drug blockade to resume abnormal cell proliferation, leading to drug resistance and tumor relapse. And a major question in the cancer field is, how does this happen? So in one model of acquired drug resistance, an initially drug-sensitive population of cells non-genetically adapts to drug to survive and even proliferate in the presence of drug. And this act of going through the cell cycle and specifically undergoing DNA replication enables some cells to acquire a bona fide drug resistance mutation, which then will lead to a drug resistant population and tumor relapse. So a lot of effort has been put into sequencing these late stage relapse tumors to understand the genetic mutations that make them resistant to the drug. But in this project, we are specifically interested in the quick non-genetic adaptations that cells might undergo just in the first few days of drug treatment, with the idea that this could represent the inception of drug resistance. Okay, so to study these processes, we have heavily invested in new single cell technologies. So first, we do a lot of sensor design. And in this talk, you'll see a live cell sensor for CDK2 activity, which has really been a gold mine because it allows you to read out proliferation versus quiescence at the single cell level over time. We also have pushed the limits of long-term time-lapse microscopy. So we can image cells in 96-well plate format with 4 movies per well, with 4 sensors per cell. So these are 4-color movies, and our current record is 12 days of straight imaging and cell tracking, which you'll see later in this talk. Of course, this generates a ton of data, so we've also invested in automated cell tracking, and we have a new cell tracking pipeline that just came out last year called ElipTrack, which is a machine learning-based cell tracking pipeline that is optimized for these high-throughput, multicolor long-term time-lapse movies of hard-to-track cells. And here's an example of melanoma cells being tracked by EllipTrack. So each nucleus is fit with an ellipse, and this allows us to handle cell-cell overlap, which is a real problem with cancer cells. So altogether, these technologies allow us to do several useful tricks. For example, we can synchronize cell cycles computationally rather than chemically. We can map a fixed cell stain at the end of the movie back to that cell's history to understand the relationship of the past to the present. And of course, we generate the longitudinal data that is really useful, for example, for developing cell lineages. So here's a lineage that goes over 5 days in melanoma cells. So before I show you a movie of the melanoma cells adapting to drug, I want to just quickly go over how the CDK2 activity sensor works, which you'll be seeing. So it consists of a CDK2 substrate tagged with a fluorescent protein. So when CDK2 activity is low, the sensor fluorescence is in the nucleus. But as CDK2 activity builds up, the sensor becomes phosphorylated. This unmasks a nuclear export sequence and masks the nuclear localization sequence, and the sensor translocates to the cytoplasm in proportion to CDK2 activity, which means that in an image, you can read out kinase activity as the cytoplasmic to nuclear ratio of this fluorescent signal. So after mitosis, CDK2 activity turns off in cells that exit the cell cycle or go into G0 or quiescence, and starts ramping up as soon as cells commit to the cell cycle. So here on the left is a movie of untreated A375 cells expressing the CDK2 activity sensor, and we're going to follow the cell with the yellow arrow. And you'll see that untreated cells cycle very rapidly. Each red dot is our automated detection of anaphase. And after about 60 hours, you'll see the cells are so overgrown, we just can't track them anymore. On the right are the same melanoma cells treated at time 0 with dabrafenib, which is one of these clinical BRAF inhibitors. So this cell completes the cell cycle it was committed to when drug was added. It divides, and it enters this prolonged quiescence for the next 4 days. And this is exactly what the cell should do because we have cut off its major proliferation pathway. But now I will replay the movie and we will follow a different cell. This one starts off just the same. It divides right around the same time, and it similarly enters quiescence. So this cell did respond to the drug. It was not a priori resistant. And it does stay quiescent for some time, but at hour 90 in drug, if you look up at the movie, you'll see CDK2 activity build up, and you'll see this cell divide into two daughters right here. So we became, of course, very interested in what was unique about this red cell that managed to escape drug compared to the black well-behaving cell. So what we do is take our many single cell traces and color code them red if CDK2 activity is high and blue if it's low, and then each of these becomes a row in a heat map. So here you're looking at 1,800 single cells that we've tracked for almost 5 days. Drug is added at the black line. And you can see the behaviors are more complex than that initial movie suggested. But in general, you can group the cells into two basic categories. So in the bottom, we have cells that divided once and sometimes twice in drug before entering a long-lived blue quiescence that lasts till the end of the movie. Whereas on the top, we have cells that do respond to the drug initially by entering quiescence, but escape at least once to divide in drug. And when you integrate over 4 days, the fraction of cells that has escaped at least once is actually fairly high, about 43%. So we thought, okay, well, maybe let's just try a higher drug dose and see if we can get rid of these cells. But what we found was that even at this very high dose of dabrafenib, we do reduce the SKP population down to 10%, but we cannot fully eliminate it. We also wanted to test whether perhaps any of these cells had any pre-existing genetic resistance or epigenetic resistance mutations. So to test that, we use this Fuji sensor, which in this case, the cells will light up blue when they're in the cell cycle and not blue when they're out of the cell cycle. So we start off with primarily cycling untreated cells, and then we treat the cells with dabrafenib for 3 days. And most cells go into quiescence, but a few blue ones are still cycling. Then we use flow cytometry to sort the geminine positive, so the blue cycling cells, SKPs, from the non-SKPs, and plate them down into the dish and give them a 24-hour drug holiday where they can begin proliferating again. And then we're going to treat them a second time. So if the initial ability to escape was driven by pre-existing genetic mutations, then when we sort those SKPs and treat them again, they should proliferate much better than the non-SKPs. But if it's a non-genetic, rapidly reversible effect, then those two populations should look the same. So we filmed the cells, and this is how untreated cells grow pretty rapidly. These are just 3 replicates. And here are cells responding to dabrafenib for the very first time. And then we can look at the sorted SKPs compared to the non-SKPs in red and blue, and we see that they fall all right on top of each other. There's no difference between the SKPs and the non-SKPs upon the second treatment after this drug holiday. So this argues for the ability to escape being reversible upon drug withdrawal and therefore not genetic. So to understand what was driving these SKPs, we use single-cell RNA sequencing. So on the bottom left, you see the untreated cells, and in the top right corner, the treated cells. And a major challenge in our field of single-cell biology is mapping between 2 different techniques. So for example, long-term time-lapse microscopy, where you can track cells over long periods, but you can only measure a few signals in a single cell, versus, of course, single-cell RNA sequencing, where you can measure many, many things, but only at one time point. But in this case, we could map between these 2 technologies because we knew the SKPs were cycling, and so we can light them up with the cell cycle gene signature and see that they fall in this little pink peninsula. So we wanted to look at genes that were upregulated in SKPs compared to non-SKPs, and in SKPs compared to untreated proliferating cells. And we found, of course, a number of pathways. And I'll just talk about 2 that we have heavily validated and that we think are very important. And they're actually both stress-related pathways. So the first is the ATF4 integrated stress response. And the second is the p53 pathway. So for anyone not familiar with the integrated stress response, this is a pathway that detects a number of different cell stresses: nutrient limitation, unfolded protein stress. Each stress activates a dedicated kinase, which will phosphorylate a translation initiation factor to suppress global translation, but it will actually activate translation of the ATF4 transcription factor to upregulate stress-related genes. So what we first wanted to check is whether indeed ATF4 protein now is induced by drug. And indeed it is. Here you can see in pink relative to DMSO. And furthermore, when we split this violin into SKPs in orange and non-SKPs in blue, we see an even more significant upregulation of ATF4 and SKPs. So if ATF4 is important, what happens when we knock it down? So here we're looking at the fraction of SKPs, percent of SKPs marked by phospho-RB, and we can see that between control knockdown cells in blue and cells where we've knocked down ATF4, we get about a 50% reduction in cells escaping in the fraction of SKPs. And this actually seems to be largely driven by cell death. So normally we get about 18 to 20% of cells dying at 1 micromolar dabrafenib at 4 days. But when you simultaneously knock down ATF4, that shoots up to about 55%. So from this, we can say that ATF4 is promoting the survival of these SKP cells that are attempting to cycle in the presence of a drug that is trying to block them from cycling, which you can imagine is stressful for cells. We also looked at a number of the top ATF4 target genes. So here's just one, CDC42EP1, which you can see lighting up here in this SKP peninsula. So we validated that by RNA FISH. So the yellow dots here are the mRNA for this gene, and pink marks the cells that are proliferating. And indeed, upon 3 days of BRAF inhibitor treatment, we see a strong upregulation of CDC42EP1 mRNA that is significantly enriched in the SKPs, just as the single-cell RNA sequencing predicted. And then when we knock this gene down, of course, we see fewer mRNAs, but also we see fewer cells escaping. So This is suggesting that not just ATF4, the master regulator, but specific ATF4 target genes are supporting cells that are attempting to cycle in the presence of drug. So could SKPs be the seed population that drive eventual acquisition of drug resistance mutations? Well, for this to be true, they would have to be more likely to acquire mutations, and they would have to outproliferate non-SKPs. So to test the first point, we remembered, of course, that the p53 pathway was upregulated uniquely in the SKPs. So we measured DNA damage using this gamma H2AX antibody as well, and then we also stained cells for DNA in blue and EDU to mark S-phase cells in pink. And we saw very little DNA damage signal in untreated cells, but this signal is upregulated after 3 days of treatment, and it's upregulated fairly specifically in the 3 cells that happen to be escaping and in S-phase at that moment. So this is quantified here for thousands of cells, and we can see that the SKPs have higher damage signal compared to the non-SKPs and compared to untreated proliferating cells. We're now trying to understand where this damage is coming from, because of course, BRAF inhibitors are not designed to be mutagenic at all. And one thing we figured out so far is that these cells seem to be undergoing DNA replication stress, which is a result of stalled DNA replication forks, which can be marked by this protein FANCD2. And again, we see low signal in untreated cells, much higher signal in treated cells, and that signal again is enriched specifically in the cells that are escaping at that moment. And it's not just DNA damage marks, but actual DNA breaks, which we can measure with this COMET assay, and we see increasing breaks, fraction of cells with breaks over time. So based on this, we can say that SKPs are prone to DNA damage and therefore will be more likely to acquire over time potentially drug resistance mutations. To test the second point about SKPs outproliferating non-SKPs, we performed a 12-day time-lapse movie with cell tracking, which was a feat only possible because of ELIPTRAC. And we saw that, okay, so first we film the cells for 4 days and classify them before into SKPs and non-SKPs, and then we keep filming. And we saw more proliferation in SKPs than non-SKPs, albeit with long periods of quiescence in between. So these cells don't suddenly become fast cycling once they escape once, they enter more of a slow cycling state. And this is maybe even clearer when we plot each mitosis as a black dot in the 2 populations, and we see about 3 times more mitoses in the initial drug SKPs. We also looked over longer terms by FACS sorting SKPs in column 3 compared to non-SKPs in column 2 after 3 days of treatment, and followed the same well over 4 weeks. And again, we saw higher proliferation in the SKPs. So this checks the second box as well, that SKPs outproliferate non-SKPs over extended drug treatment, and suggests they indeed could be driving eventual drug resistance. So one last point is that this is not specific to A375 cells treated with dabrafenib. So here I'm showing you several other BRAF inhibitors in yellow and green, MEK inhibitor in blue, and even a combination dabrafenib and trametinib, which is used clinically to stave off drug resistance. And indeed, when we use the combination, we do see a reduced fraction of cells that are still cycling after 96 hours of treatment, but we cannot eliminate this population. This is true for other melanoma cell lines like WM278, as well as for short-term cultures of ex vivo patient biopsies represented here by these 2 MB cells. So in all cases, no matter how high of a drug dose we use and no matter what cell line or patient biopsy we use, we can never eliminate all the cycling cells at any time point. So to summarize, I told you about ELIPTRAC, this new cell tracking pipeline for long-term live cell microscopy. If anyone is interested, we're happy to help you get started with it. With this pipeline, we were able to find that subsets of melanoma cells can rapidly rewire to escape MAP kinase pathway inhibition and re-enter the cell cycle in just a matter of a couple of days. These escapees experience heightened DNA replication stress and DNA damage, yet they outproliferate non-escapees, suggesting they could be a seed population that enables drug resistance. So a remaining question is just how widespread are these phenomena of rapid drug escape and associated DNA damage? Could this be happening for multiple MAP kinase pathway inhibitors in multiple different types of cancer? If so, I think this could inform the way the field thinks about the origins of drug resistance and about how MAP kinase pathway inhibitors are achieving their therapeutic effect. And these are questions we're interested to follow up on. So in closing, I'd like to thank Chen Yang, who's pictured right here, who led this project initially, and then she was later joined by Chengzhe Chan, who also worked on, was the lead author on the ELIPTRAC work. Tim Hoffman then joined, as well as undergrad Nikki Jacobson. I'd like to thank funding from the Searle, Beckman, and Kimmel Foundations for this work. And I'd also like to note that we have an open postdoc position for just the type of person who would attend this meeting. And we also have 2 new grants to fund follow-ups to this work. So if you're interested, please shoot me an email, or I think there might be some more one-on-one sessions later on today if you want to talk one-on-one today. So thanks a lot. Happy to take questions.
Thanks a lot, Sabrina, for this wonderful talk. There's a question here from Siwan Zhang. Is SKP phenotype spatially dependent? Does initial cell density matter?
Yeah, so we look for spatial effects, and in our setup, we did not find them. So we look to see, if neighbors of SKPs are more likely to escape or not. We did not find that in our setting, but that's not to say in some other setting you wouldn't find that. We do all of our imaging so that the cells never become confluent, because of course confluence will push them out of the cell cycle as well, and then you have a confounding effect. But yeah, if the cells are very packed in and you hit them with drug, they actually do better. So there is some dependency on confluency.
Great. Another question is from Jeremy Purvis. Exciting work. For the cells that escape drug treatment and show increases in CDK activity, do you know whether they have skipped mitosis or whether their daughter cells eventually arrest?
Yeah, so they don't usually skip mitosis. They normally divide into 2 daughters, like the one cell I showed in the movie. If cells are going to die in drug, it is usually in mitosis where they kick the bucket. Of course, mitosis is a pretty challenging process for cells. The second part of the question was, the daughter cells. So they're, the daughter cells of SKPs, they do, and after escaping and dividing, they do go back into quiescence for some time, but they are much more likely to escape again than a cell that has never escaped, than a non-escapee. So they don't start cycling rapidly, but if you watch them over 12 days, they are more likely to cycle again. And that's why we think they will eventually outgrow the non-escapees and why they could be this seed population for drug resistance.
Great. Okay, another question from Julia Wang. Very cool work. The SKPs are a very small percentage of the cells. So she wonders how come ATF4 knockdown induces such a dramatic effect in drug response.
So the SKPs, by time-lapse microscopy, so if you just take a snapshot time point, the fraction of SKPs at 3 or 4 days is something maybe around 17%. It depends on drug dose. But the longer you watch them, the more cells have escaped at least once. And those cells, we now have started doing, adapted a live cell sensor for ATF4. And basically, upon drug, all cells induce ATF4 to a kind of low, moderate level. But it seems like the SKPs are the ones that are inducing ATF4 much more strongly. And we're interested very much in the timing of ATF. So does ATF4 high ATF4 induction follow cell cycle entry? Like, is it a consequence of now struggling through the cell cycle, which is, our hypothesis? So I think the cells are, already partly relying on ATF4 just from the, general stress of drug treatment, but the SKPs really rely on ATF4. So that would be my answer.
Cool. Arjun Raj says, awesome work. To what extent would the rate of mutation itself be increased? Like, how many more mutations do you get per subdivision with, say, a twofold increase in DNA damage?
Yeah. So we have not measured rate of mutation. That is always tricky. We're thinking of using one of those, systems where you have something that's not a dysfunctional GFP that if you repair, if you have one mutation, or you could have a GFP that gets broken with a mutation, what the rate of that is, and kind of incorporate that into our time-lapse microscopy to get a sense of the overall rate of mutation. The other approach is like whole exome sequencing of single cells, but we haven't gone down that route yet. So right now we don't know, but we do see actual breaks by COMET assay. So in a significant fraction of the cells, that's of course the primarily the SKP. So I would say it seems to be significant, but exactly what the number is, we don't know yet.
Okay, let's take one last question from Shauna Cooper. Very cool work. While this was conducted on flat imaging conditions, do you think the SKP behavior are going to be similar in 3D conditions, where cell connections are different and drug levels are different depending on where the cell lives within a tumor?
Yeah, so one direction we'd like to go into is to grow these in a 3D culture. So you can grow melanoma cells in these spheroids. So we've dabbled a bit with that. Of course, the time-lapse imaging becomes much, much, much more challenging. Well, especially primarily the cell tracking becomes much more challenging. But with the right fluorescent sensors that turn on an escapee situation, maybe we don't have to track every cell. We can just fact sort at some time point and count the fraction of cycling cells. So yeah, that's something we are moving into in the next phase of this project. But obviously, the 3D situation is very important.
Excellent. Thank you very much. So we're going to go into the coffee break now, and we'll come back in 15 minutes. Thank you, everybody.
Okay, hi everyone. Welcome back from the break. We're going to continue on with this session. And first up, we will have Hernan Garcia from University of California at Berkeley, and he'll talk about dissecting transcriptional dynamics in development, one burst at a time. So go ahead.
All right. Well, thank you so much. And thanks to Arjun and Shalef for organizing this great meeting and for inviting me today. Again, it's a great honor to be here. And The work I'm going to talk to you about today was spearheaded in my lab by Megan Turner and Nick Lammers, but it's also the result of a great collaboration with Chris Wiggins at Columbia and with my colleagues Mike Eisen and Xavier Darczak. And the overall question that we're interested in is how do you go from a single cell to a multicellular organism? And we asked this question in the context of the fruit fly. You can see here how the nuclei in the fruit fly are going through multiple rounds of synchronized division. And at this point, all the cells look the same to you, yet each one of them knows what they're going to be when they grow up. And this is so because they are expressing a unique combination of transcription factors. And these decisions then are manifested by these massive rearrangements, for example, called gastrulation in this case. Now, the cool thing is that we know the identity of the transcription factors that endow cells with their identities. Not only that, we know that they're organized in different layers with patterns with increasing sharpness. There's a first layer of activators and repressors expressing shallow gradients that leads to genes expressing broad domains that gives you genes in 7 stripes, that gives you genes in 14 stripes that ultimately segment the embryo and think of the vertebrae of your vertebrae as your segment and endow the segments with an identity. Are you going to be a wing segment or a leg segment, for example? We don't only know the identity of these transcription factors, we also know the regulatory connections. So this is an amazing state of the art to start asking the types of questions that we're interested in. Yet I have a complaint. And that complaint is that just like this is not a pipe, this is not a fly. And what I mean by that is if I change the names in this network and I give it to the world's experts on flies, they would have a hard time telling me that this is going to give you a fly. You cannot calculate a fly out of this network description. And what I'm going to try to convince you of today is that the reason you cannot do that is because we're missing information about space, time, and concentration in these network descriptions. So our objective is to endow each one of these arrows in these networks with molecular and quantitative information that allows us to reach a predictive understanding of developmental decision making. Basically, can we predict developmental phenotypes from the network topology? And so to be more specific, what we're after is understanding or predicting how the DNA sequence gives rise to the functional relationship between input transcription factor patterns and output gene expression. Okay. And now you might say, well, this is crazy. Like you're never going to be able to reach this sort of understanding just because biology is too complicated. And I would say that's fine. You know, that's a challenge. But all I can say is that the one time that we tried to do this in bacteria, it worked. Okay. So here, this is an example unrelated to flies. This is from bacteria. This is the lac operon, where I'm going to show you some work from my PhD, where we looked at how lac repressor represses polymerase, so it doesn't allow polymerase to bind to the DNA. And in this context, you can think of different regulatory knobs that you could tune to test your models, such as the binding energy of repressor to the DNA, the number of repressors, and the number of promoters. And you can imagine it's not the same to have 100 repressors and 1 promoter than to have 1 repressor and 100 promoters. Here, I'm showing you the gene expression as a function of the number of repressors, as predicted by our theory, as you tune these different regulatory knobs. The point here is that these are 0 parameter fits. There are no free parameters here. Then we went and took the data, and to a large extent, we showed that we could predict this output. Not only that, all these curves look like different adventures in and of themselves. But it turns out that that's because we hadn't found the natural variable of the system. If you plot this data against the fugacity of the repressor, which is something equivalent to mapping a non-ideal gas onto an ideal gas, what you show is that all the data collapses onto the same master curve. Okay. So again, the point is that this took 10 to 15 years to get to that point, but at least in this particular example, this sort of predictive understanding that I'm advocating for is doable. And of course, you can ask, well, you know, this is for bacteria. Eukaryotes are a lot more complicated. How are you going to deal with that? And I agree. There have more bells and whistles, maybe. But the question is which bells and whistles do I need to account for in order to reach this sort of predictive understanding? So for example, in this simple view of an input-output function, maybe what you want to know is not only the concentration of the transcription factor, but you want to know how many transcription factors are bound to the DNA. Maybe you don't only need to know what the pattern of gene expression looks at the macroscopic level, you also need to figure out how individual cells are expressing RNA in order to generate these patterns. So the talk today is going to be divided in 3 short parts. And it's going to also be very much inspired by this great article by Joel Cohen called Mathematics is Biology's Next Microscope, Only Better. Biology is Mathematics' Next Physics, Only Better. So in my lab, we're very curious about how we can use thinking to see things that you couldn't even see with the fancy super resolution microscope. So I'm going to introduce you to experimental, to theoretical, and computational microscopes that allow us, for example, to uncover that the expression dynamics underlying these patterns is characterized by transcriptional bursting. We're going to then use theoretical models to try to predict how the binding of transcription factors regulates this bursting. And finally, I'm going to introduce you to some very new technology that we're trying to develop in order to actually correlate experimentally the binding of transcription factors and the resulting transcriptional output dynamics. And the case study we're going to focus on for most of the talk is probably the most understood pattern of gene expression in all of development, which is stripe 2 of the even skip gene. The stripe is controlled by 4 transcription factors, 2 activators and 2 repressors. Initially, only the activators are present and they give you a broad domain of activation. But later on, the repressors come on and they restrict the domain of activation into this little stripe of about 6% of the embryo length. Now, the thing to keep in mind here is that all the data we had so far came from pictures like the one I'm showing you here, which are in situ hybridization measurements, which measure the amount of accumulated RNA in the cytoplasm. Okay, so the first question that you might want to ask is, how do single cells regulate their transcriptional dynamics to result in this macroscopic pattern of gene expression? And to do that, you need to be able to look at transcriptional dynamics in single cells in a living. And to do this, we use some technology that we developed in my postdoc lab with Thomas Greger at Princeton, and that was also developed in parallel in Natalie Dostatny's lab at the Curie Institute, using technology invented by Rob Singer. And the idea is the following. We're going to add a little sequence called MS2 to the gene of interest. When that sequence is transcribed, it's going to form a little loop that is recognized by a binding protein fused to GFP. The result is that in every nucleus in your embryo, you're going to get a little spot of fluorescence, and the fluorescence of that spot is going to be proportional to how many polymerase molecules are actively transcribed. This is what you get when you put it all together. You see in red the DNA and in green the RNA signal. Now you see this broad domain of activation that I was telling you about, and as development progresses, you're going to see how the domain gets narrower and narrower. The other thing to notice as you look at this movie is that you're staring at an in vivo single molecule experiment, because these spots are reporting on the decisions made by an individual promoter in a living multicellular organism, which means that you can zoom into one of these nuclei and count the number of polymerase molecules transcribing that particular locus as a function of time. And what these measurements reveal are these punctuated dynamics that look like trapezoids. Okay, so what do these trapezoids tell us? Well, you need to remember that this technology is measuring the number of polymerases on the gene. Presumably, what the transcription factors are doing is regulating the rate at which you load polymerases onto the gene. These two quantities are related, but they're not the same. And you can relate them through a very simple model where you say that initially your promoter is in the off state, then you turn it on, and you start loading polymerases one by one, and your fluorescence increases. If the first polymerase makes it all the way to the end and falls off, but you still load polymerases from the other side, now you're going to have a constant fluorescence level. And finally, once you turn off the promoter, you're going to lose polymerases and fluorescence at the same rate that you loaded them. What I'm trying to tell you here is that this data is consistent with our model where the promoter was on for a small period of time, these so-called transcriptional bursts. And people have seen them, as you know very well. People have seen them all over the place in bacteria, in yeast, in mammalian cells. They seem to be the rule rather than the exception in biology. And the simplest theoretical model onto which you can project this sort of data is the so-called two-state model, where what we're going to posit is that the promoter can exist in 2 states on and off and can switch stochastically between those states. And only when in the on state, it can produce RNA at a given rate. So if you can measure the rate of polymerase loading, you're going to see that you could measure the burst length, burst frequency, and burst amplitude that is determined by the model parameters. So given that we know that these cells are bursting, how do they regulate this bursting dynamics in order to realize this average pattern of gene expression in the cytoplasm? If you go through the math, what you get is that this mean rate of transcription is given by a term that tells you talks about the fraction of time spent in the on state that is multiplied by the transcription rate when in the on state. What this tells you is that you can realize a stripe of gene expression by upregulating K on in the middle of the embryo, or the stripe, sorry, downregulating K off, or upregulating R, or any combination thereof. So the question then becomes, what is the strategy at play in stripe formation? And to answer that question, basically you need to measure K on, K off, and R. Now, this would be trivial if I can measure the promoter state. I would just measure burst size, frequency, amplitude, and I'm done. The problem is that what's accessible to me is the number of polymerase molecules on the gene. And you need to do some sort of inference. And this is easier said than done. And maybe I'm slow, but we spent like 8 years trying to figure this out. And only once we started working with Chris Wiggins did we figure out how to do this inference. So for 8 years, we were sitting on terabytes and terabytes of data that we didn't know how to analyze. But now we can do it. And let me show you how this works. This is basically, I would call this a computational microscope that uses hidden Markov models. And I'm not going to go through the math, but we can talk about it afterwards. But here's the idea. This, what you're seeing here is a typical fluorescence trace we get. And you will be hard pressed to tell me when a promoter is on and when the burst ends. But now we can find the most likely trajectory of the promoter throughout the whole time course. Okay. And we can do this for every cell throughout the embryo. Here we're color-coding this nuclei according to whether the promoter is on or off. And what I want you to notice is that even in the middle of the stripe, where you have the highest level of expression, you still get bursting. There's no such thing as being on all the time. Okay, so now that we know K on, K off, and R, we can measure it as a function of position, and we can ask, how does the burst frequency vary as you move along the stripe? And what you see is that, sure enough, the burst frequency is modulated in a stripe-like fashion. If you look at the burst duration as a function of position, you see that this is not modulated in any way. But the burst amplitude is modulated also in a stripe-like fashion. So what this tells you is that these 4 transcription factors that are talking to the stripe are somehow talking to some combination of K on and R, but not talking to the K off in any way. And this is more or less consistent with what most people have seen in lots and lots of organisms. Okay. In fact, I would claim that we've gotten pretty good at figuring out which bursting parameters are being regulated. However, I claim that we still have no idea about how this regulation is implemented at the molecular level. What I mean by that is, what happens? How does transcription factor binding trigger and regulate transcriptional bursts? And you can imagine a bunch of different scenarios. These are not mutually exclusive in any way. Where, for example, the transcription factor could have to be there for the whole duration of the burst, or the transcription factor only needs to be there at the beginning of the burst to prime it or something like that. So clearly you need to know something about the binding and its relationship to bursting if you want to make progress towards uncovering the molecular mechanisms of bursting control. So this is hard to do experimentally, though we're making some progress. So first we decided to put on our theorist hat and use theory as a microscope. And the simplest model that I could think of is one where we associate the on state with a state where the activator is found. And with the activator unbinds, then you don't have bursting anymore. So the simple model predicts that the burst frequency is going to be given by the activator binding rate, and the burst duration is going to be given by the activator residence time. So this is a very simple prediction. I know how to measure burst frequency, bursting dynamics. So now I just need to measure binding and unbinding of transcription factors in vivo and compare the 2 timescales. And we did this using an actual very fancy microscope that was built by my colleague Xavier Darzac, this lattice light sheet microscope. And we looked at the binding of picoid, one of the activators of even skip stripe 2. What you see here is a nucleus in the fly here. There's another nucleus, there's a bunch of nuclei here. This little bright spot corresponds to a picoid molecule being bound, okay? And I don't know if it's going to play that well in the video, but hopefully you can see some flashes of light. Each flash corresponds to a binding event. And what you can do then is ask, how long do these flashes live for? And that's a proxy for the picoid residence time on the DNA. And what we find is that that's about 1 second, which is a lot faster than the burst duration of 5 minutes that we saw. Okay, so there's a disconnect between the timescales. And this disconnect is not exclusive to binding. Here I'm showing you on the top the dynamics, the timescales measured for bursting in multiple organisms. On the bottom, I'm showing you the timescales of the many biological processes, molecular processes, that you could have blamed for bursting. And what you can see is that most of these processes are faster than the actual bursting dynamics that we observe. So there's some disconnect here. There's something that we're missing. And I'm happy to talk more about the theory in the questions, but there's one complaint that you could raise from the type of testing of the theoretical model we did with lattice light sheet microscopy, which is that when we measure binding, we're measuring binding at a random place in the genome. It could be picoid could be bound non-specifically, specifically. We don't know what it's doing. But the prediction of our model really has to do with what's happening at the promoter, where you can have multiple binding sites, when you can have cooperativity, and where you could also see what's happening with the rate of transcription at the same time. So great progress has been made on this front in yeast and in mammalian cells, but it's been tricky to do this in flies. Okay, and we have theoretical reasons to believe that at the site of nascent transcript formation, you will get long-lived events. And we can talk about it also in the questions period if anybody's interested. Okay, so how do you actually then go from just measuring binding at a random location to correlating binding and transcriptional output? And So this has led to the development, or to our initial development, of a new microscope that is based on just trying to be a little bit smarter about data analysis, not necessarily getting a better microscope. And to illustrate that, I'm going to switch gears. I'm going to show you a different arrow of the network diagram. The change doesn't really matter, but I just need to tell you some of the names of the molecules, okay? Instead of a stripe, we're going to look at a domain of this gene called SNIL that is expressed along the dorsoventral axis of the embryo. And this gene is activated by the gradient of a transcription factor called dorsal, okay? And if you take data with the MS2 technology, you'll see more or less the same type of dynamics, right? You see the broad domain appearing at the bottom of the embryo, which corresponds to the ventral side of the embryo. So you can try to measure then dorsal venous concentration and SNIL using MS2, okay? And the first question we ask, based on some really cool fixed tissue data by Ido Golding and Jun Ma, is whether I could see dorsal venous molecules binding to the DNA, to the transcription locus. Basically, would that lead to a higher concentration of dorsal at the site of nascent transcript formation? If you look at a snapshot, the answer is no. This is dorsal concentration throughout the center at the site of nascent transcript formation, and you don't see it. No matter where you look, you don't see anything, okay? Now, the cool thing is that once you start averaging over a few frames, a signal emerges. So you can see how, on average, there is indeed more dorsal at the site of nascent transcript formation, at its target site, than anywhere else in the genome. And it's a concentration that is higher by a factor of but it's like 30% or so, which by our estimations, we're talking about a few molecules, maybe 10 or so. So it turns out that there seems to be a dynamical signature to this enrichment, to this local enrichment. Check it out. So this is the dorsal enrichment dynamics at Hunchback, which is a gene that dorsal doesn't talk to. Okay, so in principle, this is our negative control. This is the dorsal enrichment dynamics at SNIL, at the locus that it targets. And you see repeatedly these little fluctuations that seem to be higher than what we see for Hunchback. Okay, so one of the things we're exploring now is whether these transient fluctuations actually correspond to unbound and bound to binding events, because they last 5 minutes. So this would be very interesting if these are actually the functional binding events that are leading to bursting. And obviously, once you ask this question, you should also ask the question of whether these binding events also correspond to bursting events. Okay, so great. So this is sorry, just got the 2-minute warning. It threw me off. So this is basically where we want to go. We want to be able to measure this enrichment, which we think is a signature of binding, and correlate it with the output transcriptional bursting. And to me, this is an experiment that I really didn't think we could do we would ever be able to do. And now I'm getting very excited, and probably a big part of my lab is going to switch to trying to think about this, because I really think that this type of experiment can reveal the so-called anatomy of a burst. So we can go beyond just saying there's a burst and how is it regulated? Is a burst amplitude, duration, or frequency, to actually knowing what are the transcription factors doing before, during, and after a burst. You can even ask, well, what are pioneer transcription factors doing? Do they need to be bound there all the time or just before a burst? And there are even ways to do some really cool stuff where you could imagine measuring the dynamics of histone modifications during, before, and after a burst, basically on spatiotemporal scales that are just not attainable by genome-wide studies. And this is only possible because we're putting together these different microscopes, okay, in quotes, to reveal the in vivo biochemical information that we need to derive governing equations of cellular decision-making. So just to wrap up, I told you that we're trying to endow the network arrows with molecular and quantitative information that allows us to reach a predictive understanding of developmental decision-making. We're hoping that this will lead us at least to be able to predict how different basis elements of the developmental process are put together, how you make gradient steps and stripes. I showed you how you make a stripe. And obviously one big question is it true that I can take everything apart and put it together, take it apart in these little basis set and put it together to actually predict how a full multicellular organism is made. So with that, I need to acknowledge these guys that I already introduced you to at the beginning of the talk. And my lab in general. It's a fantastic group of people to do science with, and these funding sources that give us the freedom to do crazy stuff. And everybody for your attention, and I'll be very happy to take any questions. Thank you very much.
Wonderful talk. I have a question from Arjun Raj. Love the Markov scope. Question, does the inference require a simple two-state promoter model? How would the inference change if one incorporated additional features like post-burst refractory period?
Yeah, so no, it doesn't. In fact, what I didn't tell you is that when we're looking at a little spot, there's actually two spots on top of each other, because that's how the sister chromatids stay with flies. And we're actually fitting to a three-state model. So you can change it to do fancier things. The question, though, that we've been struggling with is, how do you do model selection? Okay, how do you know which model is right? And we tried to do that for different states. And depending on which measure of model selection you wanted to use, BIC, AIC, cross validation, you got a different answer. So we're still not entirely sure of how to deal with knowing which model is the right one.
Okay, next question is from Jason Hughes, who asks, can your model be used to interrogate the molecular mechanism behind these gene regulatory events, such as DNA looping or transcriptional condensates, et cetera?
Absolutely. I mean, first of all, from the hidden Markov perspective, one thing is that we're starting to explore is you don't just get burst frequency, but you might get a distribution of burst frequencies. And that already gives you an extra dimension onto which you constrain models of gene expression. One thing that I didn't tell you is that dorsal seems to also show up in clusters, and the clusters move around about the nucleus, and they actually seem to be closer, on average, closer to the site of nascent transcript formation than to non-target genes. So it would be interesting to also correlate bursting with the arrival of these clusters.
Okay, Hernan, I want to ask a question of my own. So you talk about burst times that are about 5 minutes, and in principle, this could be shorter than the loading time. If you think of the length of genes and the speed of RNA polymerase, it might take like 10 minutes until it's completely covered before the first RNA polymerase falls off. So do you sometimes get these kind of scenarios, and are you thinking of integrating that into the model?
So you mean that the burst might last longer than the actual time that a polymerase lasts on the DNA, or the other way around?
So the other way around, that the burst is shorter. So the promoter it closes before the first RNA polymerases reach the end of the gene.
Yeah. So
you're not getting to the plateau.
Yeah. So the whole point of the hidden Markov model is that it deconvolves that contribution. That contribution, the memory of the RNA polymerase on the gene, is what was killing us. And that's that's the whole point of what the hidden Markov model is doing. Now, it requires you knowing the elongation time pretty well. So we've been developing some autocorrelation approaches based on some cool work by Antoine Coulon and others to measure that elongation rate. And I can tell you that there are subtleties, like, we're assuming that the moment the transcription is done, the polymerase falls off, but, termination doesn't happen right away. I mean, there are some subtleties here that as we get fancier and fancier with our experiments, we'll have to figure out.
Thanks.
I also had one question of my own. So what is the best way to prove that this increased transcription factor binding that you see by the time averaging is real binding to the correct site? Is it possible to do it in, like, film the cell and then do some sort of biochemical measurement in that very same cell where you catch it in the act of this time averaging localization? Is that feasible?
Like a single cell ChIP-seq?
Yeah.
Yeah, I think that might be tricky. I think the best bet we have is we have controls that I didn't show you, where we show that dorsal doesn't bind to other, you don't see the enrichment signal in non-binding guys, in non-target guys. And also another experiment we're doing is trying to change the number of dorsal binding sites to see whether the signal correlates with, presumably there's something about occupancy here, right? Eventually the sequences matter. I mean, even if you believe in condensates doing something, like, things need to bind to the DNA. So I think that might be the best way to do it. I don't know how to marry this with genome-wide technologies, just because the timescales are completely off. If the thing stays for 5 minutes, but it only shows up every 5 minutes, it's going to get diluted out so much when you average over the whole embryo and over all time points and such. Sure. But I would like, if somebody has an idea, I would love to hear how to do that.
Okay, let's take one last question from John Murray. At the risk of opening a can of worms, could the longer duration dorsal binding event be a signature of some type of phase separation type event?
I don't really know. Though, as I said, we see that dorsal, not all transcription factors do this, but dorsal lives in little clusters. So it could be that these enrichment events that we see are the dorsal clusters crashing against the transcription site. So we don't see these clusters forming and like fusing, but we haven't been able to image it for too long. But there is one hypothesis is that the enrichment events are the result of the transient visit of one of these clusters to the transcription site.
Awesome. Thank you very much, Hernan. We're moving
Thanks,
on
everybody.
now. Thanks. Next speaker is Gitika Arekatla from ETH Zurich, and she'll tell us about optogenetic manipulation, reviews ERK and AKT signaling dynamics required for ESC differentiation. Thank you, Gitika.
Perfect. I hope everyone can hear me and see my screen. I want to thank the organizers for giving me the opportunity to present my work on ERK and AKT dynamics in ES-cell differentiation. So to start off, I would just like to briefly say, okay, why do signaling dynamics matter? Well, in 1995, what people did was they stimulated these PC12 cells with different growth factors, which correlated to different kinds of ERK dynamics, either sustained or transient. And this was then linked to different cell fates where sustained ERK led to differentiation. Then 20 years later, people showed that it's not really about the growth factors and whatever secondary effects they may have, but actually about the ERK dynamics itself that controls cell fate. So signaling pathways can encode differentiation cues. And if you just looked at this single point in time, you would miss that and think that, oh, ERK activity over time has basically no link to cell fate. That's why it's important to follow it over time. Specifically, we were interested in looking at ERK dynamics in mouse embryonic stem cells because this is just a very simplified version of the ERK signaling pathway that lies downstream of the FGF receptor, and in the presence of a ligand, the receptors dimerize, triggering the downstream cascade till ERK gets activated. And in embryonic stem cells, if you basically inhibit this pathway, either directly or indirectly, you can keep the cells permanently in pluripotency. However, less is understood about how cells interpret different ERK dynamics and trigger differentiation. And that's where we came in and were interested in finding it out. So what we did was we introduced this optogenetic version of the FGF receptor where we removed the extracellular part, but instead linked the intracellular part with a blue light responsive protein. So that in the presence, when you switch on blue light, you actually trigger the dimerization of the FGF receptor, similar to if you have added a ligand, except now you're activating the cascade with blue light, so you have much more temporal precision over ERK dynamics. And not only were we manipulating ERK, we were also interested in visualizing the response to this manipulation. So we introduced this ERK sensor, which is fluorescently labeled. And when it's inactive, it's in the nucleus. But when you activate it, it translocates to the cytoplasm. So we wanted to simultaneously manipulate this pathway and also read it out at the same time. And that's exactly what we did. You can see here, this is the ERK sensor. And when we give pulses of blue light, we could introduce oscillatory ERK dynamics to the cells. Whereas if we gave sustained light, we could give constant ERK dynamics. And the cells behaved very synchronously. And this was very nice that we could stimulate different ERK dynamics with different light stimulation patterns. And it's not just those two patterns. We could also, based on how we gave the light, have broad oscillations or oscillations of high frequency. And also if we decrease the intensity of light, we could also have like basically the same pattern, except that we could lower the strength of ERK activation. So with this optogenetic tool, we had high control both over the duration of ERK activity and the amplitude or the strength of ERK activity, which was very nice. So, okay, it's cool that we could simulate these different dynamics, but ultimately we wanted to understand how cells understood these dynamics and how they differentiated based on what dynamic they received. And if you follow a signaling pathway over time, it has many properties, right? There's the duration of the signal. So is that what matters to cells, that a signal is on for a long period of time? Or is it the strength of activation, which is the amplitude? So if you hit a certain threshold of ERK activation, the cells differentiate. Or is this like an integral of the two, which is the area under the curve? And that's what we were interested in finding out. Because we were using optogenetics, we could actually put the cells into different stimulation programs where we could separate the different properties. So in this group of stimulation conditions, we could keep the duration of ERK the same, but change the strength of activation and thereby change the area under the curve. Whereas in this group of stimulation conditions, we kept the amplitude of the ERK activation the same, but changed the area under the curve by changing the duration. And when we linked this to the percent of cells that are differentiating, what we found was that duration or amplitude alone is not sufficient for efficient differentiation, because if that was the case, then these three conditions would have the same rates of differentiation. And if it was just about the strength of activation, then these four conditions should have the same rate. Instead, it's really about the area under the curve, where increasing the area under the curve linearly increases the number of cells that exit pluripotency. And we really looked at a lot of different stimulation conditions where the sole criteria was that over a 24-hour period of time, they should have the same ERK area under the curve. So they could have sustained ERK at low amplitude or at high amplitude with different pulse durations. And we found that it really did not matter what frequency of ERK they got, or if they got sustained versus oscillatory. As long as the area under the curve was the same, the rates of differentiation were the same. Of course, this then prompted us to think that, for all the different kinds of pulses we are giving them, the cells seem to be remembering the previous pulse because they are integrating it over time. Because in the off time, if they forgot about the previous pulse, then by the end of the 24 hours, the rate of differentiation should be the same as the no light condition, which it's not. So we were interested in finding out, okay, keeping the same area under the curve, how far apart can two pulses be where the cells lose memory of the previous pulse? So for that, what we did this experiment where we had pulses of either 6 hours or pulses of 3 hours. And for the 6-hour pulses, we gave them a gap of either 3 hours, 6 hours, 9 hours, or 12 hours. And for the pulses of 3 hours, we gave a gap of either 3 hours and 6 hours. The only criteria was that the area under the curve should remain the same. And I want to draw your attention to these two stimulation conditions where you can see that when the gap is 3 hours, the rate of differentiation is normal. However, when the gap is increased to 6 hours, it's closer to the no light condition. And you see the same thing phenomena, though a bit less clearly, when the gap is 12 hours for the pulse that is 6 hours compared to 3, 6, and 9 hours. Which shows that the memory is actually dependent on the length of the previous pulse. So cells basically lose memory when the gap is twice the amount of the previous pulse, which was, interesting to find out. Switching tracks a little bit, till now, all the experiments I have shown were done in pluripotency media. So we were trying to trigger cells into differentiation in a media which essentially wanted them to be pluripotent. However, we also did these experiments in the basal media, which is much more permissive to differentiation. And we measured the ERK area under the curve for all the different stimulation conditions and found no difference between the basal media and the pluripotency media. However, for the different stimulation conditions, when we actually looked at the number of cells that are differentiating, the percentage is much higher in the basal media, which is permissive to differentiation. So from this, what we concluded was that, okay, just having the high ERK is not sufficient. Something else is causing the difference that should explain the difference between the differentiation potential of cells in these two different media. And that's when we looked at AKT signaling, because in literature, while it's known that FGF is directly upstream of the ERK, with AKT, it's a bit more confusing, whether it's indirectly activated or directly activated. But because we could trigger the FGF response and instantly measure with a sensor, we could look at the primary responses of FGF activation on AKT. So this is the AKT sensor. And when we triggered it with light, you could actually see the sensor move out of the nucleus into the cytoplasm, showing that, yes, it is directly downstream of the FGF receptor and is also activated by it. And when we looked at the AKT response in the two different media, for all the different stimulation conditions, what we found was that the area under the curve for AKT is much higher in the pluripotency media compared to the basal media. So from this, what we basically concluded was that, okay, it's high ERK is definitely necessary, but low AKT signaling is also important for efficient differentiation. And so I would like to finally conclude that, okay, in this talk, I showed that very precise ERK dynamics are possible. We could manipulate and simultaneously measure these ERK dynamics. The duration or amplitude alone are not sufficient. Area under the curve matters. That ERK memory can be retained up to 2x the length of the previous ERK pulse, and that low AKT signaling is also contributing to differentiation. And with that, I would like to thank the members of my lab. We are in Basel, a very nice city, though not so much maybe in Corona times, and previous lab members, Andreas, who helped validating the sensors, and Christoph, who helped with building the optogenetic lid that we use for all the stimulation programs. And with that, I can end my presentation and take questions. Thank you.
Very nice. So there's a question here from Merit Romeke. Cool story. How and how long after light stimulation are you measuring the exits from pluripotency?
24 hours after. So we are constantly stimulating, and at 24 hours, we fix the cells and stain them for pluripotency factors.
And Sylvia Santos asks a technical question. Can you define what you mean by pluripotency media? Is it 2I-LIF?
No, it's AI-LIF. I hope that it's also a defined media, but it indirectly abrogates ERK activity, not directly like 2I-LIF, where you put the MEK inhibitor in there.
Okay, there's another question from Michael Peplenbosch. With the optogenetic activation of the intracellular FGF receptor, you will not only activate ERK and AKT, but also other downstream pathways of this receptor. Have you considered measuring, e.g., calcium fluxes, TARC activation, STAT phosphorylation, etc., at the same time, and seeing how they influence the ERK-AKT-mediated cell fate decision?
Yes, I mean, in the end, there are like 30 signaling pathways, and they are all of interest. For this story, we stopped here, but definitely the idea is to also look at other signaling pathways.
Okay, so next, Angus McNichol says, great talk. Does nuclear phospho-MAPK correlate with area under the curve and pulse times?
Yes, we definitely did phospho-ERK staining to show that it does correlate, yes. However, whenever you're doing antibody stainings for phospho-MAPKs, the quantification will not be as exact as with a sensor. But yes, we could show that it correlates.
Okay, there's a few questions I think related here on phosphorylation. So John Murray asks, very nice talk. Based on the time scale, do you think ERK memory is due to pathway, component phosphorylation, etc., or could there also be a role of changes in expression level? Also, Wendy Wang asks, would you expect phosphorylation status to have an effect? Is there a way to track that for both ERK and AKT? If not through a sensor, by terminal time point analysis to see if there are differences?
Sorry, I don't.
The question is, can you measure the phosphorylation level of the AKT and ERK?
Measure the phosphorylations
And if in addition to the phosphorylation levels, you might also see changes in expression of ERK in terms of the memory of the protein levels.
Yes, I see what he means. We could do that. It's just that when you're doing phosphorylation levels, you're either doing Western blots, which are again like a large group of cells, or you're doing immunostaining, which is not the most sensitive assay. That's the only thing. Yeah.
Okay. I want to ask one question of my own. So you showed that the percent of cells does not depend on the frequency or amplitude, which is the area under the curve. I'm just wondering, do you think there's going to be, like, if you took these cells and performed the RNA sequencing, ones that are undergoing oscillations or constant activation with the same area under the curve, do you think there might be genes that individual genes that respond differentially to frequency versus amplitude?
Yeah, no, that's the main question, right? So with this study, I really wanted to treat the cells as a black box and not think about transcriptional programs and really look at the signaling pathways. But yes, the next step would definitely be to look at the RNA-seq and see if basically the same genes are being triggered, and it really does not matter, or if there are two different clusters, but somehow they still lead to differentiation as the end goal. Yeah, that would be the next step.
Okay, I think we will stop there and move on to the next talk. So thank you. And the next talk is Federico Gaiti from Weill Cornell Medicine, who will be talking about deciphering the epigenetic encoding, heritability, and plasticity of transcriptional cancer cell states via single-cell multi-omics. So take it away.
Hi everyone, I'm Federico, a postdoc in the Landau Lab at Weill Cornell Medicine, and I would like to thank the organizer first for giving me the opportunity to tell you a little bit about our recent effort in using single-cell multiomics to define the epigenetic encoding and plasticity of cancer cellular state. So in cancer, genetic diversity drives evolutionary selection. However, in addition to the DNA, there is a whole host of heritable layers of information, like epigenetic layers, that also fuel cancer evolution. And so the study of cancer requires the integration of multiple heritable dimensions at the resolution of the single cell, which is the fundamental unit of somatic evolution. So to pursue this integration, we develop a single-cell multiomic platform that captures genetic, epigenetic, and transcriptional information within the same cell, and previously applied it to chart leukemia evolution. In this work, we have shown that epimutation, the stochastic heritable DNA methylation changes, can serve as a molecular clock. As cells divide, they accrue more and more error in copying their epigenome, similar to the process of genetic diversification through genetic mutation accumulation. And so this notion motivated us to use the stochastic heritable DNA methylation changes as native barcode to directly chart in human patient sample the lineage history of the cell, similar to the transformative work that allows lineage barcoding in model organisms. However, we now wanted to leverage this multiomic platform to inform the question of transcriptional cell state diversity in cancer. The notion that cancer are composed of cells that are diverse in their transcriptional state has been reported for many years. If we think, for example, about axis of cell-to-cell variation, like epithelial to mesenchymal transition. However, recent single-cell RNA-seq in the effort in the field have shown that this phenomenon is ubiquitous across many cancer types. And so cancer are made out of cells that are transcriptionally heterogeneous, often in a way that is independent of genetic clonal diversity. And so this prompts key fundamental question in cancer biology. How are these transcriptional cell states encoded epigenetically? Are they heritable? And can we learn something about the transition dynamic of this cellular state? Human glioma serve as an instructive model to start addressing this question, as cellular state diversity is an important disease hallmark with malignant cell recapitulating trajectory of neural development. And so we team up with Mario Suva lab at MGH and the Broad Institute and apply our single-cell multi-omic method to 14 primary glioma patient sample, both IDH mutant gliomas and IDH wild-type glioblastoma, or GBM. We first separated malignant from non-malignant cell based on both gene expression and DNA methylation clustering, allowing us to work purely with the malignant cellular population, which would be far more limited using bulk approaches, as well as develop a method to infer copy number aberration in this malignant cell based on the single-cell DNA methylation data. The resolution afforded by this method was far greater than single-cell RNA sequencing inferred copy number aberration, resulting in the ability to capture focal changes, as shown here, for example, for the oncogene EGFR, which would not be possible by simply using single-cell RNA sequencing. And this approach also allows us to understand genetic heterogeneity. For example, we identify 4 genetic subclones marked by either complete or partial chromosome 6 loss in different spatial regions of the same tumor. They were also associated with different level of DNA methylation compared to non-malignant cells. So now our multi-omic dataset provides a unique opportunity to directly examine the epigenetic encoding of this heterogeneous cellular state. And so we first showed that our dataset can actually recapitulate the diversity of cell state that were previously reported, spanning stem, progenitor-like state, and more differentiated state associated with astrocyte-like or mesenchymal-like products. Then the direct comparison of the DNA methylation profile across cellular state revealed that polycomb repressive complex 2, PRC2 target, are hypomethylated in stem-like state, leading to the activation of important transcription factor and HOX gene that are specifically under the control of PRC2. We validated this finding using single-cell attack sequencing data, where we also showed the genomic regions that are typically bound by PRC2 are also more open and accessible in this stem-like state. And then to examine this association in a larger sample cohort, we also look at bulk TCGA data, where samples having higher stem-like cell proportion also show greater hypomethylation of PRC2 target. Now, going to our second question, what is the heritability of this transcriptional cellular state? And this question is of clinical significance, as heritable expression program may be related to non-genetic mechanism of therapy of resistance in cancer. So to inform this question, we retrace the evolutionary history of glioma cell using the stochastic heritable DNA methylation changes, as we have done previously in leukemia, orthogonally validated by projecting subclonal copy number information that was excluded from the DNA methylation base tree building. We then further projected cellular state identity onto these lineage tree maps, and then quantify heritability of cellular state directly in this patient sample by simply asking whether cells in close proximity on the lineage tree were also more similar in their transcriptional state compared to cells that are more distant on the lineage tree. And we observe this decreased transcriptional similarity between cells as a function of their lineage distance. We then apply also Moran's I, which is a classic measure of spatial dependence to study pattern of cell state gene expression across the lineage tree. And while in the majority of IDH mutants samples, cells of the same state occur closer together on a lineage tree than expected by chance, suggesting heritability of this transcriptional cell state, in GBM sample, the distribution of cell state appear to be more random, suggesting that relative to IDH mutant tumor, GBM cellular state identity may be less stable over time or less heritable. And so this finding finally prompted to ask at which rate do transition between cellular state occur. So to answer this, we borrow a mathematical framework from quantitative ecology used to estimate migration of different species across niches. We adapted this maximum likelihood model of binary state speciation and extinction to now estimate migration across cellular state. In our case, differentiation from stem-like to a differentiated state, or de-differentiation from a differentiated to a more stem-like state. We validated this by comparing model estimate to cell cycle rate derived from the single-cell RNA sequencing profile that were not used in the modeling, and also by comparing to de-differentiation rate estimated by RNA velocity. Using this framework, we derive cell-state transition dynamics from the inferred lineage trees. And in IDH mutant, the model predicted a low rate of de-differentiation compared to stem-like cells self-renewal. In contrast, GBM samples show a significantly higher rate of de-differentiation compared to IDH mutant, allowing a replenishment of stem-like cell through de-differentiation as well as self-renewal. Thus, IDH mutant glioma resemble more the tree on your left, likely being a product of a standard unidirectional developmental hierarchy. While in IDH wild-type GBM, we see this lower degree of cell-state clustering on the tree, reflecting likely a more plastic bidirectional differentiation hierarchy. So overall, this work dissect the epigenetic underpinning and dynamic of cellular state that drive human gliomas. And importantly now, this type of data allow us to start engaging with more clinically relevant questions, such as, for example, is the high degree of de-differentiation in GBM offering an additional mechanism by which this tumor could replenish their stem-like cell compartment under a therapeutic pressure? And so shall we therefore start thinking about targeting specific cellular state transition to skewing the distribution of cellular state within the tumor? And more broadly, the data presented here show that single-cell multiomic of human sample can help linking together transcriptional cell state diversity with fundamental evolutionary properties such as heritability and transition probability, providing also novel framework for the study of human somatic evolution with broad applicability across cancer types. And so with this, I would like to conclude and thank again the organizer for giving me the chance to present our work, and then everyone that helped me throughout this work, my mentor Dan Landau and the lab, collaborator and funding from NIH and American Society of Hematology. Thank you for listening and happy to take any question.
That's great, Federico. Thank you very much. There's a question here from Noah Greenwald. Really awesome that you're able to get such fine-grained CNV call from the methylation data. Why is the resolution so much better than single-cell RNA-seq data?
Yeah, so first of all, so basically we think because the maintenance methylation machinery has an error rate which is estimated to be 4 orders of magnitude higher than the one observed for, let's say, DNA replication. We think that the phylogenetic information content of single-cell DNA methylation is actually higher than that, for instance, of single-cell nucleotide variant or other type of assay. And related to RNA-seq, also because with DNA methylation, we can rely on the concept of molecular clock, because we know it's a very faithful mark across cellular division.
Next, we have a question from Christoph Torkenzi, who asks, how much is the microenvironment in a tumor affecting cell state transitions?
Yeah, that is something that, short answer, we haven't quantified it yet, but it's something that we are thinking about and is definitely on our to-do list to dig into next. And now there are wonderful single-cell technologies that allow us to assess that, like more like spatial, transcriptomic, and similar. So it's on our to-do list to explore the relationship between plasticity and dynamics with the microenvironment. Yeah.
Great. We have another question from Arjun Raj. Wonderful work. We've also found various factors that can affect transitions in and out of these states, but we haven't found any smoking gun factor that seems to control plasticity in general. Do you think this behavior is fundamentally multifactorial, or do you think, e.g., PRC2 is a dominant player?
Yeah, so that's a sort of the type of question that we are trying to answer as well. So I think we've been seeing that there are some somatic mutations that might skew preferentially towards some specific cellular state, like, for example, EGFR might favor more the astrocytic-like state, but definitely it's not the only cause. So I think what we are trying to add here is the multi-factor layer, which is also we need to consider other non-genetic mechanism, and I think ultimately is the interplay between genetic and non-genetic that, and also again as previous question, the tumor microenvironment that might actually shape the dynamics of the tumors. So I am thinking more as a multifactorial rather than one, but we've been shown that PRC2 is one of the major non-genetic contributor, at least for the stem-like state, to keep being able to maintain their stemness potential, basically.
Next, we have a question from Jali, who says, great talk. Did you construct the lineage tree using DNA methylation data and compare them to single-cell RNA sequencing and CNV lineage trees?
Yes, so the lineage tree were all reconstructing using exclusively DNA methylation data. And what we've been doing is then like leveraging the match single-cell RNA-seq to then project the cellular state identity, because again, we think that the resolution that we can get from single-cell DNA methylation data is actually much higher than that, for instance, copy number aberration, which still they allow you to reconstruct lineages, but you wouldn't get to like probably to the resolution that we can get with single-cell methylation, rather more clonal relationship, and that's been shown in other studies as well. And yeah, so I think it's all a matter of like trade-off between how many cells you can leverage with the resolution, and I think single-cell DNA methylation is a sweet spot in that sense.
Awesome. Thank you very much, Federico.
Thank you.
We'll be moving on now to Rinat Arbel-Goren from the Weizmann Institute, who will tell us about multicellular filaments of an Avena cyanobacteria.
So first, I would like to thank the organizers for having given me the opportunity to present our study. The study was carried out in the group of Professor Joel Stavans from the Weizmann Institute of Science. As you probably know, circadian clocks can play a key role in many biological processes. Circadian clocks from unicellular organisms to animals may increase fitness by temporarily aligning biological processes with day and night cycles. A lot has been learned about the unicellular cyanobacteria in the context of circadian clock in the unicellular organism Synechococcus. And in this organism, the core circuit controlled post-translationally by three proteins, Chi-A, Chi-B, and Chi-C. Chi-C has two phosphorylation sites at serine and threonine, and Chi-C can be in one of the four phosphorylation states: unphosphorylated, phosphorylated at T, both at S and T, or only on S. In addition, Chi-C is regulated by Chi-A and Chi-B in a feedback loop configuration. Chi-C transduced its output signals down to downstream genes via the master regulator transcription factor, RPA. Some of the input signals are the ATP-ADP ratio and the redox state of the cells. So we wanted to study how do circadian clocks behave in multicellular cyanobacteria. In order to address this question, we choose to study Anabaena. This is a wonderful one-dimensional filamentous cyanobacteria organism. As you can see in this picture, this cell consists many cells, can be hundreds of cells. There is cell-to-cell communication between the cells, and therefore that suggests clock coupling and even synchronization. So not much is known about circadian clocks in Anabaena. The only study that was published about circadian clock oscillation was done at the level of bulk study by DNA array measurements. They have shown that about 100 genes exhibited oscillatory behavior, but none of the Chi genes of Anabaena displayed high amplitude expression returns, as opposed to Synechococcus. So we wanted to study a clock-controlled gene in which have high oscillatory behavior. We choose to study PECB, which codes for a subunit of photosynthetic pigment, and this gene PECB. We study that by following the expression of PECB fusion to the promoter of PECB fused to GFP, a translational fusion at a single cell level in real time under fluorescence microscope. So using we also have phase contrast images. We segmented the cell and measured the mean fluorescence intensities of each individual cell along the filament. So having this strain, we took movie. So here I show you a movie a typical movie that we took in the lab. At the left, in red, you can see the autofluorescence pigments in Anabaena. In the right, the expression of PECB from its promoter. So this movie was taken for about 5 days, every 30 minutes, in a gel pad-based device. The cells were grown under constant light condition, which suggests the autonomous behavior of the clock. And as you can see, synchronization and spatial coherence maintained despite cell-cell variability. So having these movies allows us to measure the clocks at the individual level, at the individual cell level, and to measure their lineages. So here I show you 2 cells and several generations. You can see from this figure that the phase is inherited upon cell division, and that the lineage preserves ancestor phase for several cycles. We can also measure the average fluorescence intensities of the filaments, and you can see here in pink that oscillation maintains several generations. However, when we deleted the clock genes, as shown in white, the oscillatory behavior was abolished. Note that the oscillation are abolished, or there is no autofluorescence pigment oscillation in the wild-type background strain. So in order to study the spatial correlation along the filament of PECB, we measured the special autocorrelation of the fluorescence along filament by use the autocorrelation function. So this function relates the fluorescence of cells with that of its nth neighbor. As seen here in this graph in blue, we measure the autocorrelation function of the wild-type background, and we find that it decays in about 5 cells. So we can this correlation can be due to the inheritance after cell division or due to cell-cell communication via septal proteins. So in anabayanite, it was published that septal proteins such FAST-C and D may transfer small sugars and metabolites, but not core clock components. And therefore, we developed a strain in which we deleted this septal protein. And as shown here in green, you can see that the autocorrelation function decay in a length of about 2 cells. Note that similar decay was observed in the Chi deleted background as well. So deletion of clock genes or septal proteins involved in sugar and metabolite transport reduce septal coherence. In addition, for comparison, we show in pink the correlation that would expect solely from inheritance after cell division obtained from stimulation. And as you can see, the inheritance after cell division accounts partially for correlations. In addition, we wanted to measure the synchronization in the filaments. So in order to do that, we calculate the synchronization index as suggested previously in the literature. The synchronization index is defined as the ratio of the standard deviation of the mean fluorescence intensities of the cells with time, divided to the standard deviation of the intensity of cells along filament, averaged over all cells. So this index is varied between 0, which is unsynchronized, to full synchronization, which is 1. So at left, you can see here, for example, the filament. We follow along the synchronization or along the growth of the cells, but we choose cells that are uncorrelated. So we took enough distance, at least 10 cells, between the cells that we choose to analyze. And when they grow, you see that this distance is increased. So we found that the wild type background, this index is about 0.85, which is a quite high value. However, when we deleted the septal proteins, in this background, we found that the value is significantly smaller. So disrupting cell-cell communication reduces synchronization. So to sum up, today I showed you that circadian clocks in anabaena are coupled via cell-cell communication, that oscillations are highly synchronized and spatially coherent, and for a stochastic model that includes demographic noise in phosphorylation state, see our upcoming paper. So I would like to thank the people that contribute to this study. This study was done in a group of Professor Gerald Stavans with collaboration with the group of professors Enrique Flores and Antonia Herrero, and the group of Duccio Fanelli from Florence. And I would like to thank you for your attention, and I will have a question now.
Thanks a lot, Rinat. Maybe I'll start with my own question. So there are situations in your beautiful movie where the sort of the filament folds and you have contact between far away places. So I'm wondering if synchronization is only along the line or even just adjacencies could somehow synchronize.
So the cells which are touching each other are in very long distance. The coherence is quite, we find it along the filament, but there is no cell-cell communication between these cells. It's just due to some sugars that there is contact, but there is no, we assume that there is no real transport or things that are correlate. So it's actually via the contacts between the cells, meaning channels like the septal protein. It might be others, of course.
So Jason Hughes asks, how fast do these cells migrate, or how much motility do the filaments have, as cells moving in and out around each other could alter or impact the spatial correlation? So I guess kind of related to what we were just talking about.
Okay, so the cells are just, it's like it's a filament, so it's like, there are contact like that. It's a real monofilament, so it's a filament that all the cells are connected. And therefore, the motion is due to their growth, because each time in between the cells are grow and they just become longer and longer. So it's like many cells that are in a train that every time it's split in the middle. So this is the reason that you see that, but actually the cells are in a gel pad that is between two in slide, and we keep the focus for several days. So the migration, there is no real migration, it's just the cell division. What we see. It's due to cell division, the growth of the filaments.
Right.
Okay, and maybe I'll ask one more question. Can you say something about the evolutionary advantage of these long filaments and sort of their sort of the limitations? Can they just continue to grow? Are they occasionally broken up into single cells or smaller filaments? Do they ever merge? Anything like in general about this biology of this weird organism?
Okay, so actually, if you know, Synechococcus is a single cell, but in the case of Anabaena, what is beautiful about there is no merging. Actually, what happened is that in this is very, it can be hundreds of cells. It can be very, very long. And with time, sometimes they break and they continue to grow, but they can be sometimes hundreds of cells. So it's like a very long filament. And the binary division is in E. coli. So they just divide each time, and then you have another, every 24 hours, you can see in average cell division. It's not so fast like in bacteria or in Synechococcus, so it's every 24 hours, so they have a longer cell cycle. Yeah.
Okay, so there's one comment from Arjun that got several votes, which is just a comment about how beautiful your movies are. So thanks, Rinat, and we will now move on to Stefan Rulans from the Max Planck Institute for the Physics of Complex Systems, talking about from sequence to space and time. So take it away.
Yeah, thank you very much. Thanks to the organizers for having me. In recent years, we've seen breakthroughs in single-cell genomics that allow probing detailed molecular states of cells along the linear DNA sequence. As we know, biological function relies, however, on where these molecules are located in the physical space of the nucleus and how these molecules work together on the cellular scale, such as in phase separation phenomena or in super. In this talk, I will show how such spatial temporal processes can be inferred from single-cell sequencing experiments. And I will demonstrate our approach, giving the example of the establishment of epigenetic marks during early development. DNA methylation is a primary layer of epigenetic regulation. It affects cytosins in a CpG context, and it's mediated by so-called DNMT3 enzymes. In development, DNA methylation is first erased from the paternal and maternal genomes, and then re-established between embryonal date 4 and 5 in mouse. To understand how the methylome is established, we used an in vitro model for this phase in development, where we cultured cells long-term in 2I conditions, and where DNA methylation is erased. And then we release these cells to serum conditions where these enzymes, DMT3, are upregulated. Release, we then performed a bisulfite sequencing and single-cell NMT sequencing time course over roughly 60 hours, which gives us access to the methylome, to the transcriptome, and to local chromatin accessibility in single cells. DNA methylation, as you know, is regulated locally by many different factors, and consequentially, average DNA methylation was established with different rates for different functional annotations. However, we found that actually these curves differed only by a single number, such that when we rescaled time, these curves all collapsed onto a single curve. And I think in one of the earlier talks, we already have seen another example of data collapse. Now, this is what is in physics called scaling behavior, and it means that there's only a single mechanism of how DNA methylation is established genome-wide. And we, as physicists, with our equations, we can actually hope to understand something. So now this single curve followed almost exactly a simple power law with an exponent of 5/2. In the physical sciences, such power laws are hallmarks of collective or self-organization phenomena. Now to understand whether de novo methylation is coordinated in space, we calculated so-called correlation functions or covariance functions. Previous speaker called it autocorrelation functions as a summary statistics of the arrangement of methylation marks along the DNA sequence. Roughly speaking, it gives the probability that two sites, a given distance apart, have the same methylation state. What we see is that these correlations decay much more slowly than expected by typical enzyme kinetics, shown here by the red line. In other words, DNA methylation marks are strongly correlated along the DNA sequence, reflecting potentially spatial coordination of this de novo methylation process. Now, taken together, this shows that de novo DNA methylation during development is an interesting testing ground to infer spatial temporal processes from single-cell sequencing. And this is what I'll do for the rest of the talk, to give you an overview of our approach, first using tools that we derive from quantum physics. We first infer the kinetics along the DNA sequence from single-cell data, and then map this kinetics to a description in the physical space of the nucleus. From this, we can then infer higher order or larger or mesoscopic phenomenology, such as phase separation phenomena. Now, to go a little bit into detail, as a first step, we made a general mathematical ansatz for enzyme DNA kinetics, where enzymes bind and unbind from the DNA, and if bound, they can modify the DNA, they can methylate a given position. These enzymes can also interact in general and up to no unknown ways with each other. Such interactions contain, of course, the interesting biology, and our goal is to learn them from the data. Now, the stochastic time evolution of such kinetics is described by so-called master equations. To solve such equations for general interactions between enzymes, we are able to reversely then infer the shape of such interactions from linear sequencing data. And here, which is something, of course, a complicated procedure, I just present you the result of this procedure for our experiment. What we found is that the binding rate of DNMT3 enzymes decreases with the power of 1/3 with a distance to bound sites. Now, these are the interactions. But the biological meaning of such interactions becomes clear when we calculate the total binding rate in the vicinity of a bound site. And this binding rate increases with the length of such a region to the power of 2/3. And this has a straightforward interpretation. It is the surface to volume ratio of almost any three-dimensional object, meaning that only a fraction of binding sites are accessible for further binding. Therefore, according to the data, de novo methylation relies on a positive feedback with local DNA compaction. Now, the next step to infer kinetics in physical space of the nucleus, we calculated how small length elements in physical space co-evolve with small length elements in sequence space, which we actually would be measure in the experiment. This leads then to a mathematical the time evolution of the concentration of methylation marks in the nucleus. And this mathematical description, again, that we can infer or derive for the experts in the room, as shown here, this resembles those descriptions that are used in phase separation phenomena, such that our prediction is that this de novo methylation process leads to the formation of condensates of methylated DNA in the nucleus. Now, to give you something more intuitive, we performed numerical computer simulations of this model that we inferred. And this computer simulation here that you see here, color denotes the concentration of local DNA methylation. And you can see how over time, hotspots if you go from bottom to top, hotspots of condensates or condensates of highly methylated DNA emerge. And we predict that the size of these condensates to be in the order of 5,000 base pairs or roughly 40 nanometers. And to challenge this model, we asked whether it was capable of predicting the genomic distribution of methylation marks. And indeed, this model that we infer from the data and that has actually 0 free parameters was indeed able to predict the genomic distribution of DNA methylation marks represented by the correlation function for a broad range of genomic features, as well as the spatial association between DNA methylation and chromatin accessibility that you see at the bottom. Now, to test our prediction of DNA methylation-dependent emergence of chromatin condensates, we analyzed single nucleus M3C sequencing data of serum embryonic stem cells, and we've indeed found an abrupt increase in mid-range contacts for genomic regions exceeding an average DNA methylation level of roughly 40%, which is, of course, fully consistent with our prediction of a phase transition and the emergence of 30 to 40 nanometer-sized condensates. Structures of strikingly similar size have recently also been observed by super-resolution microscopy, as you can see on the right-hand side. Finally, without going into much detail, we were also able to predict the genomic distribution of methylation marks in the embryo, so in embryo data. And in addition, using this framework, we identified specific changes in the genomic distribution of DNA methylation marks in gene bodies that precede gene silencing at the exit from pluripotency by up to 2 days. So 2 days before we see something in the transcriptome, we can already detect changes in the methylome. So finally, to come to a conclusion, I showed you that we have a general method for the inference of emergent spatial-temporal processes from single-cell sequencing. And using this method, I showed you how the establishment of the embryonic methylome relies on the interplay between chemical modifications of the DNA and dynamic chromatin structures. Finally, as a side remark, this also emphasizes what I showed here, the usefulness of analytical mathematical methods that complement the existing computational-rich computational toolbox that we have in single-cell genomics. Finally, let me thank the group of Wolf Reich at the Baber Institute and Tim Lohoff, who did these experiments, and Fabrizio Almeida, the PhD student in my group, who did this very difficult mathematics that were actually part of this that I showed you were considered to be unsolvable, and he corrected, and also the Max Planck Society and the ERC. Thanks a lot for your attention.
Thanks a lot, Stefan. We have a question from Jason Hughes. Can your approach then use linear space information to infer 3D organization, as the 3D organization would potentially determine which marks would be put on the same time, i.e. enzymes acting on DNA that are close in 3D space? The 3D organization might determine where marks are located in linear space.
Can you repeat the last part of the sentence?
So if as 3D organization might determine where the marks are located in the linear space.
Exactly. So I think, if I understand correctly, what I showed here is that you cannot really distinguish the 3D organization and what is happening in linear space. Because what is happening in linear space, in this example of methylation, is fundamentally determined or a product of the three-dimensional processes. Yeah, I hope I got this question correctly.
I think so.
Let's see. Arjun has a comment and question. He says, beautifully quantitative results. How might folding properties as revealed by Hi-C and others impact the results? Like if there's a loop, would you see deviation from the L to the 2/3 law?
Yes, that's a very good question. So these typical results that you get from Hi-C data typically occur on much larger spatial scales. And they would not be reflected in the DNA, in the statistically reflected in the data that I showed you, especially in the distribution of correlations in DNA methylation patterns. Yeah, so these processes exist also, like other enzyme kinetics, the processivity of DMT3 enzymes and so on. They probably exist. There's lots of evidence for them, but they're statistically, so to say, not the dominant process that is going on. So they cannot explain neither this, what I showed you in the beginning, this power law with an exponent of 5/2, or the shape of these correlation functions, which is the spatial distribution of these methylation marks.
Okay, we have one more question from D. Hu. Do you think these self-organizing properties could be recapitulated in vitro?
Yes, so it depends. So I showed you some in vitro results in terms that we have cells in vitro, embryonic stem cells, cultured embryonic stem cells. If you think about in vitro, like the synthetic biochemistry experiments, these have been done partially, and they showed, actually, for example, they showed that tetranucleosomes, if you methylate them, then they stick together, they have attracting forces. So there are synthetic biochemistry experiments that go in the same direction of what I showed you, which is, of course, good because that shows that it's physically possible and not against the laws of nature, what I'm showing you.
Great.
Okay, I think we will end here. So a few reminders. So the unanswered questions will be moved to the session forum, and you can continue the conversation with the speakers there. That's on the webpage. All the sessions will be available on demand on the homepage by the end of the day. And next event is the Networking Lounge, which is at 2 p.m. Eastern Time or 12 p.m. Mountain Time. And then we will come back here for the final session, which will be at 5 p.m. Eastern. So yeah, so I guess we have about an hour's break. And if you want to come back for the social networking hour, that will be at 2 p.m. Eastern. All right. Thanks, everybody.